o'clock now. So welcome this morning to the symposium. This morning's talk is all about the fourth industrial revolution and plant breeding. So we're going to start this morning with a keynote speaker with Prof. Tulio de Oliveira. He's online. Unfortunately, he could not make the symposium, so we're going to have a Zoom meeting with him this morning. He's ready. Good. Let me introduce him. It's Prof. Tulio de Oliveira. I spent the last 10 years focusing on advanced molecular techniques, such as genomics and bioinformatics. He also develops and sells genomics services and diagnostics. He enjoys supervising research teams. He currently has more than 50 people in the Genomics Africa team. Prof. de Oliveira also have published more than 150 publications and also developed plus minus 21 software applications. So he will be online joining us. So, Prof. Tulio, you're more than welcome to proceed. Thank you.
Okay. Good morning, everyone. Apology not to be able to be there with you. To be honest, I was very exciting about learning more about plant breeding and the roles of genomics for that. But I do run in our university in Durban. I'm part of the coronavirus war room. And one of the guidance that we did is people not to travel. So I have to follow my own guidance. Yeah. But today, what I will be talking to you, it is about Genomics Africa and how fourth industrial revolution technologies can and are being used to advance research development and genetic engineering in Africa. So if I just go to the next slide, I just would like you to start imagining. So we're just gonna take the next few minutes just to imagine. Imagine if you could, from a simple buccal swab, very quick identify all the DNA of the pathogens, but how also of that the individual, and you could start predicting the risk of someone having a heart attack or developing diabetes or developing cancer. But imagine that you could also use that information to derive precise prevention and treatment for the most important diseases that are affecting or that will affect South Africa. So imagine if you could do that the same, not only from disease point of view, but if you could use our whole biodiversity. As you are aware, South Africa rank, depending of the ranking, the top 5 countries in the world of high level of biodiversity. If we could use that to identify, again, new molecules for therapeutics and vaccines for both animals, humans, and plants. If we could that to already spearhead a very developed field in South Africa of agricultural biotechnology and starting advancing industrial biotechnology in the country. So imagine if in the time of mass immigration from South Africa, you could reverse the brain drain. Imagine if you could identify researchers that have been training the top American and also the top European universities and could bring them back to South Africa. But not just any researcher, but the researchers, South African researchers that have been there for years or decades, of people that's very committed to see high-level science and development happen in the country. Imagine if you could do that. Imagine if you could host this number of individuals in state-of-the-art facilities with laboratories that is as good as the top laboratories in the world. Imagine if you could host them in a R1 billion building, a building that hosts 300 full-time researchers, with also have been designed for maximum collaboration and maximum change of ideas. Imagine if you Imagine then that these people with access to the highest technology in a great environment could then be mentored by some of the brightest South African and African researchers, yeah? People as one of our fellows of the Royal Society, yeah? Slim Karim, South Africa only have 3 fellows of the Royal Society of the UK. Or another South African called Alan Aderan that went in exile, became a full professor in Rockefeller. 3 of his students got Nobel Prize winners, and he went to found what's the most advanced system biology institute. Or imagine you could be mentored by people that also brought diagnostics of vaccines to the market. Example, Professor Daria from Stanford that have created a multi-billion drug for heart disease, or Professor Michael, also from Stanford and now in Sydney, which have created the most used vaccine for chickens, yeah? So imagine if you could join them and also be mentored by some of the top African scientists, people that have developed the best scientific parks in the world, or the best genomic facility, or also some of the CFO of our top banks. Imagine what you could do that. So that's what we did at CRISP, what we founded, what we call the KwaZulu-Natal Research Innovation and Sequencing Platform. So what we did is to set up a state-of-the-art DNA sequencing laboratory in Durban, but also quite important to attract some of the brightest South African individuals that have been exiled for years or outside the country to now contribute to that. So that's what we did in Durban. And we also got this fantastic mentorship that I mentioned to you. But the topic of this talk is about fourth industrial revolution. So I would like to highlight some of the techniques that we have set up here. CRISP is slightly different from most of academic laboratories. One, we are not academic laboratory. We are a service laboratory to service both the academic, but also the private industry. And that means that our laboratories are fully ISO accredited, as would be a high industrial facility. We have invested a lot on automation, because if you were to do things at the same price, speed, and quality as the top genomic centers, you also have to use the same technology. So we did a big partnership with companies such as Perkin-Elmer to automate the whole process of Illumina sequencing. Also with Thermo Fisher Scientific to automate the whole process of Sanger sequencing. And we have bought one of the largest equipments that can do up to 3,000 sequences a day. So not only allow the throughput, but also drastic reduction of price. We have AutoContinuous, a big partnership with Thermo Fisher to also automate a lot of the big next generation sequences. And we are quite excited that last month we opened one of the first, at least to the best of our knowledge, CRISPR-Cas9 laboratory that offers service to the community, and that with all the equipment to do incubation, transfection, and visualization of genetic modification. What is very important about CRISP is that in addition of generate all this data, you also have to invest quite heavily on the analysis of data. So we have 10 full-time bioinformaticians that access very large computer clusters and that have a large number of bioinformatics software already installed and already pipeline. Yeah. Important, this is a service that's available not only to academics, but also to commercial firms to also secure proprietary data. So our bioinformatician CRISPs, we do generate data well in Genomics Africa and CRISP, but we are very good on analyzing and developing software applications. In the last few years, we have started a new field, what's called microbial genome-wise association studies. That's both for pathogens that infect humans, but also animals and plants. We got invited by Nature to write a review to present this new area. In 2018, we published a very widely used software in Nature that can do very large, big analysis because the data begin to explode. Last year, we published something that's become very interesting to this community that we call the Genome Detective Virus Identification Tool. It's a fully automated pipeline to identify all non-viral from genomic data from sequences. Yeah. Because we had these developments, it also allowed us to respond quite quick to big outbreaks. So we work around the clock in the beginning of the year to release a whole genome detective to identify and characterize coronavirus genome. And we are very glad that was made available end of January and paper was already published. Quite important for CRISP, yeah, is that we do service a lot outside organizations. A lot, as you notice, we are a lot focused on pathogens, on medical organizations. We are beginning to serve a lot of big American organizations and universities which have big projects in Africa. We commonly imported samples to over 24 different countries in Africa. We are beginning to, as the lab become accredited, to also move things for diagnostic. And to our great surprise, we see a big uptake of agriculture and conservation services. We do a lot of sequence from the South African Sugarcane Research Institute, for the Oceanographic Research Institute, but also to big conservation and agricultural programs within Africa. More important to everyone is that CRISP is not the only laboratory that has access to this facility. CRISP formed with Diplomics, with another 6 laboratories distributed around the country, a network that want to provide researchers in South Africa and Africa with high-level world-class omics facilities. So laboratories included the Agricultural Research Center, the ARC in Pretoria, the St. Jane in Worcester, the Central Analytical Facility in Stellenbosch, the MRC Genomics Center in Cape Town, the CPGR in Cape Town, and the NICD in Johannesburg. Between all these laboratories, what they have is access to some bigger sequencing technology. For example, the NICD has a very large PacBio, which we can also have access through Genomics Africa, a number of NEXT6 and Sanger sequence. The ARC has our highest high-seq in the country, in addition to a very large Illumina gene chip. The CPGR has access to a number of NEXT-seq, that's the intermediate Illumina sequencer, and also a large Illumina gene-seq. And the MRC Center in Cape Town just brought the largest sequencing machine to South Africa. It's a BGI MGI 2000, which can do whole human genomes or whole mammal genomes or whole plant genomes. At the moment, we are running the first big program on that, and we just have completed by last week the first 30 human genomes that have been produced in African soil. Another very well-run facility is the Central Analytical Facility of Stellenbosch. They focus only on Thermo Fisher technique, but they have very large servers, proton machines, which allow to do very large complete service. And they have a big interest on agriculture service. Yeah. So in addition to that, we are also very committed from Diplomics and CRISP and the other laboratories in a very large training program. We are really focused on genomics, artificial intelligence, automation, and programming. Since CRISP was founded, we have trained 1,500 individuals. And this year, we're going to be doing 16 workshops on the basics of data generation and analysis. We also took a large number of PhDs and MSCs, and we are very committed to equity in South Africa. So, but in this venue, as a keynote speech, what I want to launch is a great ambitious idea, which we call the Moonshot Project, which we think that we are getting ready to launch this missile to the moon. And that it is a program that we want to launch that we call the South African Most Important Genomes, or SAMIG for short, yeah, which are very ambitious objectives to sequence and analyze 10,000 of the most important genomes in South Africa in the next 10 years. So how this program is going to be launched is that we will ask the community 3 important questions, such as what's the most important genome be sequenced. Imagine the unit, both academic but also private in industry, will have different answers. Someone may say that the most important genome to be sequenced is the macadamia, what is a growing crop in South Africa. Other ones may say that's the coronavirus, if you are in infectious disease. So we want to ask these questions. We want to ask, why is it important to sequence the genome? Is just because it's a pet project, or is of national importance? And how does that would benefit South African human welfare and economic development? So this project will also link the idea is to run selection committees that are focused in different sections of the project. For example, we should have a South African and most important genome committee that focuses in animals or mammals, another one in plants. So they should be able to select what's the most important organisms to sequence, another one in microbes, which we will probably lead, and another one, for example, in industrial biotechnology. So the question that's come, is that kind of ambitious project, what's the benefit for the country? And has anyone have done that before? The answer is yes, that would fit a very large, big project called the Earth Biogenome Project, which the main one of the first goals is to benefit human welfare. And it's important that this is not just academic project, because the idea is not just to develop papers, but to develop new treatments for infections and inherited disease. That's on the top left, or to create new biological synthetic fuels, or to generate new biomaterials, or to generate new approach into feeding the world, or identify drugs through slow or reverse aging. That's some of the benefits. And in South Africa, we think that's very important to also focus a lot on economic benefits, as we have to kickstart our economy again. So what's the cost of such project? A very large genome at high quality that haven't been sequenced should cost around $40,000. That's for a large organism. A resequenced large genome of a plant of animal should cost around $20,000. Interesting, this price are falling every year. And a high quality small bacterial genome with diversity around $5,000, and a small genome of a new virus with good diversity around $1,000. So we crunch some numbers and we come with an initial idea how much money we would need to do such an ambitious project. The idea should divide that in phase, with phase 1 for the first 3 years, focusing a lot on samples, biobanks, some equipment, but the bulk going for sequencing and increasing costs for informatics. So our estimation is that around 2.7 billion rands will be needed to do such a project in 10 years. In the first 3 years, close to R1 billion investment. That sounds quite a lot of money, but let me tell you, this is like 10 times less what South Africa is giving to South African Airways for a year, or 50 times less from Eskom. Quite important that the idea is not going to come only from government funding, and I'm going to highlight that the next slide, but very important, it is the return on investment. For example, the Human Genome Project cost $2.7 billion and generate a return of investment of 141 times, more than 1 trillion to the world economy. So our objective is to raise funding from both government, scientific agents in South Africa and internationally, also private funding, also offered as private service as CRISP and other laboratories do, donation, crowdfunding, and philanthropic. The idea is to raise 2.7 billion rand in the next 10 years with a potential for very high return on investments. Diplomic is considering to do a seed funding with the objective of using this money to leverage more international, especially money. It's much easier to leverage international money if you see some investment from your own government. But again, that it is like 1% or 2% of the costs. Other countries are also spearheading that kind of project. They see a massive economic benefit. The UK have launched last year the Darwin Tree of Life project. They have a little bit more money than us. So the objective a little bit more ambitious, 1,000 unique species that they have identified in the UK in the Genomics England laboratories. Yeah. That project also will field and will collaborate and access data from other smaller projects, such as the Chilean 1,000 Genomes Project, a country that's also not a developed country, development count taking that, but also organism-specific projects such as the genomics for Australian plants, or for the Genomic 10K, or the BATS 1K project. So what is important is, before you go mad sequencing, to also access what have been sequenced and curate the data. So that's my last slide. So why the South African most important genomes? Why Fourth Industrial Revolution, and why Genomics Africa? So South Africa is one of the most biodiverse countries in the world. It's ranked third in some of the reports. It's consistently ranked in the top 5 or top 10 countries in the world. Yeah. We have a number of local genomic centers with bioinformatics expertise that's already in place, and they are well situated to make a big contribution to characterize the variation of biodiversity, but also important to generate that products that can develop the bioeconomy. One very important thing, that the only way that we're going to be able to do such a project in South Africa, if we produce data at the same price as the international centers. Otherwise, our samples will continue to be exported. And this is a large project that's a partnership between academic, governments, civil society, and also private individuals. So thank you for your attention, and I really hope you have heard what I talked to you. Otherwise, I am just like a crazy guy shouting to himself in his room. And I would like to open for questions or discussions. Thank you.
Thanks, Prof. Tulio. Any questions?
Thank you very much. This is Shimelus Hussein. I'm also from UKZN Pietermaritzburg campus. I'm interested to, if you can give us some highlights in terms of the application of the results of the technologies, especially the genomic edition, or the sequence data that you will be doing on those selected areas. What's really the practical use? What do you perceive from now that the data generated can be used in those areas? Say, for instance, you are doing plant genomics, some viral genomics, and animal genomics data. So what will be the area of application?
Okay, so I can highlight our initial applications. So yeah, CRISP in the last 3 years, yeah, we published 150 papers, with 20 of them in Science, Nature, and Lancet journals, that the big journals of our field. So one of the big applications was to increasing the scientific output to international levels. Yeah. The other big application that we had is that a number of these technologies are already moving through the clinical diagnostics. So for example, we have launched a very successful diagnostic for precision medicine. So that is for cancer, for diabetes, and for rare diseases, and for pathogens such HIV and TB drug resistance. So that is technologies that left our laboratory to go to the commercial field. So and that's a lot focused on virology. Yeah, on virology, we are one of the things that we are very exciting and that we are currently validated is this large metagenomic process that you can identify all viruses infecting our organism, and that's not only human virus. The other application that left the laboratory and it went to the commercial field, it is the genome detective. It was founded as a small company that does service for very large sequencing centers around the world. So that's some of the examples of the that left information. So a lot of them will focus on our expertise of virus, but what we are doing now, we managed to get some funding from the Spark Global Program, is to expand this translational science from human, but also to try to bring that to other organisms such as plants and animals.
Tulio, you excluded or seemingly excluded human genomes from your SAMIC project. Can you please just explain how that project will link to human genome projects or human sequencing, genome sequencing in South Africa?
So yeah, can you see? I didn't really exclude that. For me, humans are mammals. Yeah, so it should follow under the large mammalian genomes initiatives. Yeah. In our case, we have been the group that was the first group to generate human genomes in the African soil. Yeah, so that's a funding from the South African Medical Research Council. The first 200 genomes, human genomes, to be produced in South Africa or in Africa, they are focusing on children that's naturally protected against HIV. So that is the first big project going to the MRC Research Council, yeah. And I imagine that many more will follow about that now that we have this technology in South Africa. So it will be to the scientific community to get, you have a lot of South African human genomes that already have been published, yeah. You have over hundreds and hundreds that's mostly be done outside, but it's going to be to the scientific community now to identify other phenotypes that are important to be sequenced.
Good, thank you. Thanks, Prof. Tulio. I think that's it for the keynote speaker. Thank you very much.
Thank you very much, and apology not to be there.
So the next speaker is Prof. Renee Prince, and from Shenzhen and US.
Okay. Chairperson and committee members and attendees. I'm also affiliated, like Chris said, to the Stellenbosch University Department of Genetics as an industry partner. And as Tulio just explained, we are also included in the group of service labs of the Diplomics Initiative. I thank you very much for the opportunity to share with you the follow-up chapter of the Shenzhen story. I think many of the young scientists here in the audience were still in primary school when I gave my talk in 2008 to this audience. And
yes, I'm here.
I can just say to you that we face many challenges on this continent, but we also have many opportunities. So I hope that our story will give you hope to the possibility of a unique career path. At Shenzhen, we have a team-based approach in everything we do, in-house and in our partnerships with our collaborators across the globe. And therefore, it's maybe fitting that I forgot to add my name to the title slide. Shenzhen started off from the humble beginnings in 2003 as an owner-management business to create a vehicle to practice science when my family relocated to the rural town of Worcester. And it developed into a fully fledged business that is well known today. It provides specialized research and development services and also applied services to plant breeders, fellow scientists, and various stakeholders in the agricultural industry. It's a bit intimidating to talk about this after listening to Richard's talk yesterday of the evolution of precision breeding at Bayer, or just after Tulio's talk now. But in 2003, I was also told that it will be very difficult to establish a private lab. Unfortunately, it's 17 years later, and I still feel that we have a role to play. At SAPBA 2008, we reported on Syngene's initial focus to address the gap between the molecular genetic knowledge and its application in our local agricultural industry, excluding, obviously, the multinationals. The successful bridging of this gap in the sectors which we focused on led to increasing support from our industry partners and funders, but also significant recognition of the small yet efficient team by international collaborators and funding bodies. And I would like to highlight 3 pivotal points of the past 12 years. Oh, sorry, I didn't see it.
Much better than
Please
get
go
a
on.
plane about the Oh, hello.
Where do I
I
point?
didn't have to
Is
go
it
to the
moving
toilet,
on?
and I gave my thought.
Sorry about that.
Very well.
So the 3 pivotal points I want to highlight is maybe very fitting because I hear the load shedding schedules started again. The placement of robotic instruments and the challenge we have to equip us with new data handling skills.
Yeah, but it's like...
I just would like to touch quickly on the timeline of our exposure of integration of some of the fourth industrial revolution's technologies. In 2013, we were fortunate that Dr. Leslie Boyd, through a BBC SRC grant, she's a colleague of Eric Ober of NIAB, made it possible for us to install the first entry-line CAS platform at Syngene. At the time, it was the second system in the Southern Hemisphere. In 2015, I had to install a solar system at the lab. In 2016-17, we started with a drive to develop crop-specific bioinformatics expertise here in South Africa, hosting our first workshop in 2017. In 2018, we upgraded our CAS platform to extensively and could establish the placement of a DNA extraction robot. 2019, we hosted a follow-up bioinformatics workshop, and we built our first Linux computer at Syngene, and we just completed hosting the third bioinformatics workshop. Now, I would like to start with something very non-scientific but very relevant. In 2014, we were confronted by this concept of load shedding to cope with the shortage in the electricity generation capacity in this country. Considering many factors, including safety issues of having to refuel a generator in the middle of the night, we were forced to take the big step and invest in a solar system. Now, this highly sophisticated first world system consists of solar panels bolted with specific secure bolts to the roof so that it's not being stolen. Then it's linked to a battery bank, and it has an inverter, which is a highly specialized brain that performs and manages the energy system and decisions automatically. And this can all be followed online. However, when this system experienced its first load shedding, it actually did the exact opposite what it was intended to do and completely shut down all energy in the lab. And it was a way to protect itself from being damaged. And this was a very beginning of a long journey to figure out how a first world system can integrate with the energy supply system it has to manage, that certainly does not meet first world standards. It's working, fortunately, perfectly today, but the developers of these systems would just shake their heads if they see all the extra surge protectors and switches we had to install to allow us to manually intervene when needed. So we experienced the lack of something very basic as a constant electricity supply as a huge challenge to operate on a level where we can embrace the fourth industrial revolution. Our initial CAS platform was replaced in 2018 with a fully automated system, which also included the DNA extraction robot. To integrate these instruments in our workflow, we had extensive training, which I can tell you is invaluable. It's not just putting a PCR machine down and figuring out how to operate it. We also learned that the maintenance of these systems are very expensive, and it is advisable to rather collaborate than duplicate, if I can give you any advice. However, it took our data generation capacity to a next level, where we often find that the bottleneck is rather on the side of the breeders, who often experience capacity constraints to provide the number of samples that the system can cope with. One of the crops we work on is wheat. It has a complex genome. It's 5 times the size of the human genome. And I was trained in the era when the field of bioinformatics didn't even exist. And we are all aware of the need to be able to work with big data sets, but each plant species has its own unique challenges, and that is very difficult, and it's very difficult for scientists with no background to this field to attend a course on mosquito or human and then translate that then to your crop genomics. So I started this drive to find funding, to have expert training in bioinformatics with wheat as a model. And the first workshop was made possible with funding from the Newton Fund, and we were only, due to their rules, we were only allowed a 1.5 day of discipline-specific training, but that was just the spark we needed. On a trip to the UK, visiting their leading research institutes, the British High Commission recognized our dire need in crop-specific bioinformatics training, and they assisted us to host in 2019 a 5-day workshop. And at this workshop, we tried to raise awareness among some of our local bioinformaticians about the unique challenges we as scientists face here in the crop space, and we continue with our training throughout the year using an online platform. So it moved on. So we just completed, as I said, our 3rd course with funding that Stellenbosch University and the British High Commission again made available. And we were trained at the intermediate level now. We're not entry level anymore. And I can just say, yeah, that Sinjin took the initiative to create and secure funding for these learning opportunities to the benefit of all the serial scientists in South Africa. To integrate our newly learned knowledge, we had to get a bioinformatics server, and with expert guidance from abroad, we managed with a consultancy in total of 3.8 hours, we managed to identify and order the bioinformatics server that we would need. How much time is there? 5. Okay, so I'm just going to quickly run through what we actually do. Our interest is to identify useful genes, to develop tools for that, and to use those tools to assist other the breeders to either identify genes carrying the traits faster. And we also study some of the pathogens of some of the crops. Over the course of many years, we have been leaders in the field of identifying useful genes, mapping those genes, integrating the tools immediately in the breeding programs for wheat, barley, and also grapevine, where we collaborated with the ARC. We always try to source tools in the public domain, so we won't duplicate what's already available, but often you have to try and find markers yourself. It's a constant process. We have now decided to also integrate the more complex traits and looking at the root diseases. We subject ourselves, although we are a private company, to peer review so that we are evaluated, and we are proud of the fact that many of the tools we develop are shown on public databases as a sort of marker of choice for that gene. We serve as a central service for the commercial wheat breeding programs in South Africa, and just for our international experts, we already started in the 90s to implement marker-assisted selection in South Africa. When I attended a conference abroad and just once again saw a marker I developed being utilized by international scientists, and knowing very well it's not being applied in South Africa, we approached local funders and asked them to help Syngene as an independent lab to support the breeders to integrate DNA technologies in their work. Now we're still lagging behind. We haven't even touched on genomic selection, so that's our next aim. The international breeders are already using that, but we need some training to incorporate that and to assist the breeders with that, so that will be our next goal. Our flagship research project is cloning a few of the disease resistance genes that we're making very good progress in. We have achieved a fantastic milestones where we, as Syngene, as a company, have secured the Pioneer Open Innovation Grant. We have been successful in a internal cost grant. So internationally, we are now also recognized as a safe and a reliable research partner. We like the things we work on, a whole range of things, and I always feel a lot of satisfaction that we are involved in the agricultural industry. We couldn't have done that without all the support and many of you guys that either help us with plant material development, phenotyping, providing field trial sites. It wouldn't have been possible, and we say thank you for that, and hopefully we'll achieve more successes in future, but we are ready to take the next step because we just appointed Dr. Xolelwa Zulu as an independent director at Syngene. Thank you.
Any questions? We have about 2 or 3 minutes left.
Thank you.
Thanks, Prof. Renee. So the next speaker will be Dr. Zelda Bizet from the ARC.
Good morning, ladies and gentlemen. I've got a talk for you today on the futuristic tree. I just want to warn you, this is not a scientific talk. I thought so early in the morning and being bombarded by so many wonderful talks yesterday, let's keep it a little bit light. So what is the futuristic tree? So before I move on to that, Existing megatrends says that in 2013, there will be more people on Earth, more of whom will be living in cities. And with bigger shocks, there will be less arable land. There will be much more pressure on our water sources. Our waters or oceans will be overfished, and the water levels will be rising due to overheating oceans. But we have also learned that the fourth industrial revolution will change how we buy, how we grow, and everything that we do. So maybe it's not that doom and gloom. So what I want to see first is, what is the fourth industrial revolution? So probably you know this by now, but I've just highlighted it for us that are very biological and not really into technology. And as you get older, technology becomes a struggle. You have to ask your child to come and change the TV channels because they changed something and you don't know how to get it back. Now we sit with a fourth generation revolution, and it's challenging. But it says it's a way of describing the blurring of the boundaries between the physical, digital, and our biological world. So no more denying technology. And then also it's a fusion of advances in the artificial intelligence world. That's not just computers. It's computers that learn from their environment and then do better in future. It's actually self-programming. Then there's robotics. There's the Internet of Things. We have 3D printing where you can actually print anything that you want in a 3D form. And then also what we know by now a lot about, because we're biologists, is genetic engineering. And then also the quantum computing, and then also other technologies that it's slotting in all into this. Now, obviously, as you can see there, we are mostly concerned with something like genetic engineering, or like me, far away from the lab, more in the field. Okay, so I just want to say, what does this entail for breeding of trees? I know most of the people here, except maybe Dr. Tsele, is more involved into crop breeding, in the field crops. I think, Dr. Lane, you have been lots working with trees and fruits as well. But for most people, tree breeding is a very grey area. And I've been talking a lot about the constraints and everything. I don't want to go into that again. But I want you to close your eyes. Just close your eyes and picture a tree. Your favorite tree. Okay. And then I want to just have you picture that tree. Maybe it looked like something like that. It's a huge tree. It's a tree that's nurturing the nature around it. It's giving shadow to the animals around it. For me, my favorite tree was that one, because it had a lot of emotion in it, and it actually sheltered humans and their emotions. But unfortunately, the trees that I work with doesn't look like that, and it can't look like that, because that's not economically. Okay, so during all these years, since cavemen was there, domestication of crops started to take place. Now, we don't know really how it worked, but we can think of how it worked. We have people that at that time was gatherers. They would go in the field and they will go and pick up the fruit for themselves, choosing what they think or perceive as being nice to eat and nice looking, maybe big enough, and they will test it, and if it's awful, they will throw it away, and if it's nice, they will eat it and discard the seeds at their home, and the trees will grow. Or they will even try and plant it, and as we know, planting seeds, they might end up not having what they want. And so the trees became domesticated through natural selection, or not really natural selection, but like a selection pressure. And therefore, we now have a tree that I'm supposed to envisage. It's the one on the right-hand side. It's a nice tree, full of nice, wonderful fruit. It's not doing what nature intended it to do, because nature wants it to survive. It's doing what I want it to do. That's producing as much as fruit as it has to, so that the farmer can economically farm. So we have moved from what we have known on the left-hand side as wild orchards or woodlots or forests into what we have as monocultures, which brought along a lot of other problems. The fruits doesn't look the way they should look. I don't know whether you have seen this before, but it's very interesting to note that right at the top there's a peach. The right one, on the left, you will have your wild types of peaches. And through years in being domesticated and bred extensively, it changed to what we know as a peach now. Bananas used to have seeds. I don't know whether you know that, but bananas used to have seeds. We don't have bananas with seeds anymore, making it very difficult to breed anyway. Then the watermelon is just below the peach. It had very definite segments. Now we have fruit that has what we want, nice, lots of flesh to give our children to eat. Tomatoes at the bottom, it was little cherry types of tomatoes. It has been bred to become nice, big tomatoes that you as a consumer will walk into the shelf and say, yes, I want that one, and not that little mouse balls over there. Okay. So, and then at the top, that's my favorite one. It's the citrus. Now imagine me sending my child with the left-hand one to school and say, peel it and eat it. Yeah. Okay, I think he will also look not very happy at the end of the day. Good. So there's one factor that is very, very important that will never, ever change. When you want to improve something, there's only two things you can do. You can either manipulate the environment in which it grows, okay? Or you can manipulate the genotype that grows in a set environment. It's only the two things you can do. How you do it, that's basically where we all get different approaches and we like to argue with each other. But those are the two basic concepts. Now, in my case, manipulating the environment pertains to orchards. We plant all these trees in nice right rows. We used to plant them in squares in the old days. You could actually land planes in between them. They were 12 meters by 12 meters apart because we didn't know about another manipulation practice, which was pruning, okay? So now these days we plant them 3 by 6 or 5 by 2. They are in rows. And yeah, they must go north to south, not east to west, because our sun is moving the other way around. I don't get optimum sun. So we've actually been artificially intelligent. We have learned, like we want our computers to learn, what is the best practices. On top of that, we measure our environments, okay? And we look at what is impacting on our trees. We measure the weather, the soil, whatever we can measure, we try and measure, and then from there we deduct. I think Mr. Richard Fly had a very wonderful talk on precision farming. It's exactly that. You measure and then you react on that. When we manipulate the genotype, on my way, I love to do conventional breeding, lots of data, and then deducting knowledge from that. We've just heard about state-of-the-art genetic tools in 2 talks at this morning. And then we can also manipulate the genotype by pruning it. Remember, not the environment, then I manipulate the genotype. I can actually put it on a trellis, bend it, fold it, do anything with it. I'm manipulating the genotype to the environment I've created for it. Okay. Just to show you that even our measurements have evolved. On the top, there's a nutrient probe that you would have said, okay, that's how I measure what water is. It was a huge setup that you have to put on an orchard. These days, it has evolved to the right-hand side. We've got a little water marker, okay? And the water marker is a little thingy that you put in your soil, and you can actually go with a meter to all of them. If you're very technology advanced, you can have it transmitting all the knowledge to your office. If you're a lazy farmer, you can sit in your office and have everything at hand. No, it's not lazy, it's effective. There's just one thing. You have to get into your orchards. You can't farm from your chair. But that's all equipment that helps you to know where to get to your orchards first. At the bottom, we've got a weather station. The one we had to put up on the left took almost a whole block up. On the right-hand side is a little one that you can actually put into your tree. So you can actually measure the climate at that tree. So we see what has evolved for us. For conventional breeding, like I said, we have our whole conventional program at the top, starting with crosses and moving through the whole program. You've seen that before. We've started to manipulate our genotype. I mean, when Adam took the apple from Eve, he never thought that he could change the roots of that tree, that it has 2 different genetic parts. But we've learned that we don't have to breed everything. We can actually paste together 2 things and get a new thing that works better than just 2 one apart. Then at the bottom, we can actually just take out the gene that we want. We've learned that, so we can manipulate our genotype. Okay. So then, what's there left for a futuristic tree? What must we look at? Are we going to go on like we are doing now? Now, if you look at what is the main aim in my case for citrus farmers, it says I must maximize the long-term global competitiveness of our farmers in the world domain. I must thus breed highly stable genetic cultivars, which won't go and, falter in an environment that's different than where it was bred or where another person is already making money with it. These days, people want a one-solution cultivar. Why is that farmer getting the cultivar, not me? I can actually explain to him, your climate. No, you should have bred it that it suits my climate as well. So that's unfortunately what we have to do. But the other problem is that we are breeding what the consumer wants, whether it's good or not. The consumer wants seedlessness, improved color pigmentation, improved quality, and then also a change in ripening time. Unfortunately, high sugars, it's not very conducive to diabetics or even normal people like me. If I eat too much fruit, I promise you I will get fat because it's bred past what it was supposed to be. The sugars are way too high. Our climates, our water is under pressure. Our land use demands. I mean, you have to plant in excess of at least 10 hectares to make an economic unit on citrus. Then our earth is getting ever more polluted. No, not by the people there in townships or in cities, by our farmers ourselves. We overspray chemicals. We overuse our water. Did you know that by spraying your trees with poisons or your chemicals to take off your funguses and so on, you use more water than that tree uses in irrigation? So we don't calculate that in our water component. We forget that we put those liters of water into the tanks. So yes, maybe the relevance of our breeding objectives should be rethought in this new fourth industrial revolution era that we're moving in. Unfortunately, we will then have as breeders to influence the people out there because they will still say, I want a sweeter one. It mustn't have seeds. It mustn't look as ugly as that old peach. I want it to look beautiful. Remember, I pay a lot of money for this. I must impress my neighbor. Okay. So let's see what maybe we should look at in future in this futuristic tree. What about a sugar-free orange? Is it possible? Maybe not totally sugar-free, but by knowing what is all the chemical constituents in that fruit that actually gives it its taste. Remember, if it's just sugar, I could give you a glass of sugar water and a glass of Coke. You will know the difference, okay? You agree. Now, they take out the sugar out of Coke. You still sort of perceive it as Coke, okay? So we can lessen the sugars if we know what is the components that impresses you about that cultivar, that actually make it taste like a mandarin or an orange or a mango, okay? There are some of our superfruits that is anti-cancerous, but it is just also a group. They don't maybe have all the right components in it. It's a level, the amount that you have. We have to test that and breed it in to be higher. Then our tree forms. If we have land use problems, we might in the end up that you have small farms of less than 20 hectares that has to produce what you used to produce on 100 hectares. So maybe we should start rethinking of stop worrying about what the consumer wants in sugars and seeds and stuff, because we've been doing that now for almost 80 years. Maybe we should be proactive and start on thinking of to tell the consumer, we can give you something that's still nice and still tastes well, but it won't have that bricks. So don't go and try and taste the bricks. Just eat it first and come back and tell me if it's nice. Okay. So what about pillar trees? Where you can actually train your tree to bear more fruit on a specific small size of tree. Is that viable? This is all ideas. Remember, it's not scientific talk today. It's just opening up the minds. Let's think out of the box, see what we can do. What is the trends in the world currently? Remember, we have the orchards that we are used to as farmers. But we've seen more and more that people in the cities want to grow their own food, but they don't have areas to plant in. So they move up into their rooftops. They move into their basements. We have container farms. And then we also have glasshouse farms, and we also have, these days, automated indoor growing of vegetables, not yet fruits. So, are we going to really just go merely more efficient? Like at the left-hand side, that's hectares and hectares and hectares of area under net for water conservation. Do you know what you've created there? A desert. It's a desert. It's not being tested because agriculturists are not environmentalists. We just give what the farmer wants. Nobody thinks of that as a possible desert situation. No carbon sequestration. It's all in the net. No water leaving that system, or very little. What are we doing to the environment? Is it really more efficient long term? At the bottom, we have some of those trees that we talk about that's rolling out into the field in a trellising system. We have more fruit on a smaller area. Is it more efficient? We don't know. We still have to test that. Is it sustainable? And then for you that really did try and that really came from KwaZulu-Natal without fear, I salute you. You will recognize that structure on the right-hand side. Those are the glass houses that you are used to see at King Shaka. They are huge. You can grow trees in them. I would die for them to be a Nelspruit, okay? But I'm not moving to Natal now. No. Okay. So the next thing that's also on our fourth industrial revolution, and there we already see our farmers, our researchers, the people are proactive. There's robotics. They are actually picking the green peppers or the yellow peppers in the glass houses, which is indoor. At that right-hand side, you won't know what that is. It's not a vacuum cleaner. It's a weed picker. It actually runs through your orchard, identifying weeds and then very selectively picking out the weeds for you. It could save a lot on chemicals. And remember, when we're saving on chemicals, it's not just rand and cent. We're saving also our health or water sources. Because remember what I said, how much water we spray when we put chemicals on our trees. So wouldn't it be nice to have a robot in your orchard that doesn't tire, doesn't want to go on lunch? And that one's got a sun panel on it. Maybe at night it would like to be on a power source. Oh, just let's go back. We also get drones that can fly over our orchards and report to us, are trees happy? What's happening in the trees? We can measure more than we could ever measure in the past by sending out even 20 students in your orchard taking physical measurements. So we can get more phenotypic data, and you will hear a talk tomorrow by Dr. Made Boyse that says it's now the time to actually embrace your biometry because data are getting bigger and bigger. Okay, so farming exoskeletons, this is a new one. It was also new to me. I can't put on too many photos because they're all protected. But this is when you put on an exoskeleton. Instead of climbing onto a ladder, you just walk into the orchard, you jack yourself up, and you just take the fruit. Put it in, and then you go on, and you jack yourself up, and you get a fruit. Apparently, it's much more safer than climbing on a ladder. I'm still having my reserves on that one. Or are we going to be really futuristic? Remember I said merely efficient or more futuristic? This is like building a farm city on levels with robots and trees being grown in a very controlled environment. But financially, is it the investment worth? Or is our earth that investment worth? Still have to be decided. Is that going to happen, or are we going to sit for the next era just going on the old way that we did? Okay, next, and it's just my second last slide, nanobots. Is there anybody here that has heard of nanobots before? It's new. It's where you have maybe something like a cancer, and that nanobot can zoom in directly on that cancer. Currently, it's being used in the medical field. They still have a problem with the body perceiving it maybe as a virus and trying to eradicate it. But being a computer with AI, it can actually maybe also dodge those cells. It can't go and produce on itself. So if you put in 3, you can extract 3. Currently, they don't extract it, but you can maybe in the future have a homing device and get them out and reuse them. Probably, this will be much more less expensive to produce than any other technology that we have, due to 3D nanoprinters. Okay. Nanobots are modeled on the E. coli bacterium. The E. coli propels itself with a little flagell tail at the back. So they modeled this on that system. Now, currently, in citrus, we are already using viruses to protect our plants against other viruses. We pre-immunize, but we have to breed that viruses. So, with 3 billion nanobots that could fit into a teaspoon, it's quick to produce. Is it more inexpensive than, say, producing your viruses that you don't know how it's going to react with your plant because it's a living creature? So this is something to think about. One day, the nanobots may even be smaller. A nano is a 9th million. Atoms are much smaller than that. It can actually go and work on atom level. And that can be picking up your G, your C, your A, your T, and you actually put it on a computer, and it goes and runs and puts it in an order, building an artificial DNA. What about something like that? I said to you, it's not scientific, it's just opening up your minds. There will, however, always be the phenotype versus the genotype. No matter what we do, we still are either manipulating the environment or manipulating the genotype. So forever we will have that with us, that our phenotype is equal to our genotype plus our environment plus the interaction in between them. But we will have to rethink how many interactions we are now talking about, because it's not just your genotype and your environment. Your environment has 1 million factors now. How are we going to put that in our computing? So, is the sun currently setting on our normal view of how an orchard look, nicely on the farm, with rows and rows of peaceful trees standing there, that farmers visits very early in the morning when nobody is up already? Or are we going to see our farms in future, in cities, in buildings? It remains to be seen. All I wanted you is to rethink on what we are thinking, what we are doing with the technology is currently coming so quickly available to us that we can even print veins. We can even print plant parts. We can maybe put a nanobot in the tip of our plant that can magnetically zoom onto where it should be on the training system. No more training, no more pruning. You just program your nanobots, and your tree grow up and it just connects where it should on your training system, There's so many options. So when we start looking at breeding, we will have to start thinking on how are we going to evaluate? What are we going to evaluate? Or are we just going to go back and breed a very high sugar cultivar because that's what you want to buy from the shelf, and I'll do that until the day I die? Let's think about that. Thank you.
We have about time for one question.
Nice presentation, thank you. So from your own perspective, as you know, our society is technophobia, and the last speaker yesterday was actually talking about how is society going to perceive all these sciences. So from your own perspective, how do you project that the society will perceive the use of such technologies on natural things?
takes. The farmers will do whatever it takes to adapt, because remember, they've got to survive, just like their plants have to survive. So I don't think we have farmers that are really technology phobia anymore, okay? And also, if I look at what is happening in society, there's a drive towards healthy food currently. People are just very slow at it, and they are very demanding. It should be healthy and beautiful as well. So it takes me to now try and get healthy, beautiful, environmentally safe, farmer satisfactory, all into one cultivar. But like I said, it's not always necessary to breed everything. We breed the most important things. And whatever we can manipulate, and it's cheap to manipulate, rather manipulate it. If it's a thing that you can quickly and easily do by gene editing, let's do it. There is still, however, a huge phobia out there about GMOs or genetically changed food. People are moving currently, it's also a trend, towards raw foods, natural things, wild stuff. Not that it's also healthy, that's not to say, but that's a perception that's out there. So yes, I think maybe the technology, the young people, not maybe me, the younger generations, I mean, you give somebody an iPhone, he's 3 years old, he doesn't have a phobia for that thing, I promise you. And he'll know by much quicker than you how to get to his shows on that thing. So I think it will evolve, yes.
Thank you, Dr. Zelda. Our next speaker will be Tasha Rasmus from SAPBA.
You see so many familiar faces. I think in the agricultural industry, you always encounter the same faces again. They might not always work for the same companies, but be for sure at an event like this, you'll see them all again. So it's nice to see familiar, friendly faces when you stand in front like this. I need to give you a little bit of perspective of where we come from. SAPBA Laboratories was founded in 2008, really, as part of Incotec. We did a successful management buyout in 2016, which allowed us to get a more holistic approach on what is happening in the industry. That's why it's so important for me to be here today. It gives me an opportunity to see what's happening and to tell you all to keep doing what you're doing, else I can't do what I'm doing. We've become involved in the whole cycle of food production or crop production, from the soil fertility, which we're collaborating with Eurofins Agro. They are sitting in Wageningen. They've given us the opportunity to offer a service to look at soil fertility, at all the nutrients that's needed by different crops. Soil samples and water samples are taken throughout the crop cycle. The irrigation water is analyzed, and we look at the nutrients in the leaf samples of the plants as well to make sure that during the growth cycle of the plant, they've got everything that they need. To take you back to our original slide, we are involved in physical seed quality testing. We've had an international audit on the 25th of November last year to become an ESTA accredited laboratory for Southern and SADC countries. We've been recommended to receive our accreditation and waiting for the final documentation, which is a great highlight for us. The normal seed testing apply here. We focus mainly on physical seed purity and germination. With the addition of our ESTA accreditation, the services will be more. There's a sampling service that gets added to the current services. And we have expanded the sites that we cover in South Africa by also having another site up here in Pretoria. So we have a Pretoria branch that performs physical seed analysis and the one at head office that does exactly the same. Besides the physical seed quality testing, the reason why we're here today is the plant breeding, and that is where we started from originally, offering a service as plant breeder support for the small-scale farmers, for people just starting off. What is important for us is that we do not attempt or start any service unless it is accredited or in collaboration with an international partner. We've received registration from DAF as a biotechnology facility that allows us to test GMOs. The GMO testing is internationally accredited through ISO 17025. I'll tell a little bit more about that in a moment or so. The normal goes. A lot of breeding practices, plant production, seed propagation. Things go wrong. We offer that service to assist in genotyping, to do variety comparison, variety identification. We have launched an embryo rescue department or tissue culturing department that also does meristem propagations to ensure virus-free plants. These are services that we have brought into play to assist and to offer a toolbox for the breeders, which is all of you out there, to make your life easier. We don't all have access to the technology that the multinationals have, and we have to start somewhere and give some kind of support. By incorporating your toolbox, or the tools in the toolbox, we can accelerate the breeding process to a minimum of 3 seasons in comparison to your traditional cycle in a season. I do want to say a little bit about our general diagnostics program that we're running for Clearfield. We are a BASF approved facility to offer the assistance in Clearfield sunflower breeding as well. And then we have one of the phytosanitary tests that we perform for government, looking at PSA in rice samples. We do offer funded research projects. We are not a research company. We do piggyback on what you're doing, and what we're good at is to commercialize your ideas. Make it work where it counts, and then that's another tool for the next generation to use. You can all read, so I will not go through that. We can ask some questions in the end. To complete the little circle, I need to tell you a little bit more about our flagship services, which is GMO testing. We have been accredited since 2010 to perform GMO testing. We do this now in collaborations with Eurofins GeneScan, that's another Eurofins company. We are also distributor for Eurofins GeneScan into Africa, a major challenge for us because we're actually scientists, not distributors. But we use the technology, and we are busy to applying for Eurofins certification to assist with trade conversions, which is a great need in the industry. Currently, as a routine test, we do adventitious presence testing, which combines both qualitative and quantitative analysis of different traits. We stay abreast of what is happening by looking at what is new, what new events are being released, and there we take our guide and our cue from what is happening in the rest of the world and with the technology developers. To enable us to retain our accreditation, we perform annually 4 to 6 proficiency tests on all the certified or accredited methods that we have in our laboratory. And as another part of breeding support or breeding services, we offer trait homogeneity. So everybody here is aware that most of the African countries are opening to transgenic plants, or transgenic, it's not a good word, I've been told, but to use technology in Africa to speed up their breeding processes, and we are there to support that initiative to do the trait conversions. Finally, we added, to complete the circle and to reap the reward of what you are all involved with, is to breed food, ultimately, that can be consumed safely by the consumer. So besides looking at the soil, making sure that the seed has got good quality for selling and exporting, we ensure that the food that will be on your table is not going to harm you or kill you. So we look at mycotoxins, which you know is a major problem in grains and cereals. We ensure that there's no pesticide residue on the final product, and we check for food allergens. These are all, besides for the pesticides, there are a lot of breeding that can be done towards the prevention of food allergens. This we have a joint venture, or a unique partnership with Eurofins Dr. Specht. They are based in Germany. All of these things we started based on a demand out of industry. Knowing where we came from, where our roots are sitting in molecular breeding and breeding support, we understood the importance of making sure that what we start with is also attractive to the consumer in the end. By performing these tests, we can ensure that there's less recalls of products. The environmental hygiene is up to scratch. We all had a huge big scare with the Listeria outbreak last year, which also shook up our little company into a new avenue of testing and making sure that what we do right. By getting involved in this and understanding the full cycle of where we start from the soil, growing the plant, breeding the plant, selecting the right genotypes, phenotypes, everything that the consumer need, we are able to complete our little circle of what we offer to the client or the consumer in the end. There are some bad stuff that we had to learn to do, which we have never been exposed to. We had to learn how to test for food pathogens, which gave us a bit of a scare, because when you are dealing with plants, you are never really aware of what else is happening out there. In the last year, we had another 8 methods accredited to allow us to perform or complete the cycle that we have been involved with in producing from the soil to your plate, to make sure that what we are doing here is actually coming into the industry and reaping the benefits of everybody's efforts in this room. So the final bit that we added was our food pathogen testing. We are in talks for additional collaborations. Up to this point, we have been drawing a lot of attention from Eurofins, which is an internationally based company, and we will be partnering with them for additional services that will improve and also speed up the work that we are doing to support the plant breeding industry in South Africa. And then I've made up a lot of time, I think, because it went really fast. Thank you for your time. Thanks.
Any questions?
Nope.
Thanks for your talk. So I'm just interested in your GMO testing. Is that based on ELISA, on detecting the protein or detecting the DNA when you're detecting BT or Roundup Ready?
Real-time PCR.
Is it all based on real-time PCR?
Yes.
And so do you work on what threshold can you detect? What is the level that you actually need to detect in a sample?
Our cutoff level is 0.05% for detection and 0.1% for quantification. And in terms of copy numbers, that means it's about 20 copy numbers that's a legal limit of detection. Different countries we've seen have got different parameters. In Africa, of course, it has to be below 1%. Europe is completely closed, but there are still the people that thinks of all the technology to test for. And we're constantly adding to new and lower detection limits. But at some point, it's not realistic anymore.
Okay.
Yeah. Anyone more? One or two questions more?
Thank you. Also on the GMO area, with the new plant breeding technologies where you can edit with CRISPR 1 or 2 nucleotides, in South Africa, law at the moment is still considered to be GMO until released. How are you guys going to test for that? Do you see that as a possibility to test? Because how are you going to say, oh, this was naturally bred versus lab manipulated?
I think that would be really hard. We have had a lot of discussions on that. If I can use an example, on Clearfield Plus, that's a single nucleotide polymorphism. So it will probably go the same route. Okay. Thank you.
Thanks, Tasha. Our next speaker will be Dr. Shamay Naidu from SAPBA.
Okay, good morning, everybody. So I'm going to be talking about how we used QTL data in our hybrid breeding program, our eucalyptus hybrid breeding program, to actually make selections. Okay, let's go. All right, so I think let's just start with what is the basics of any breeding program. We start with a parent population. We then make selections, often identifying the best individuals in the family, and then we then plant that out and we want to move the mean of that population. So in this study, what we want to do is we want to add in the genetic component. So how can we make selections based on the genome of those plants? Okay, so this study was part of a big project which included the genomic mapping of both growth and wood properties in conjunction with Professor Meyerberg's group, the FMG. And the aims were basically to use DNA marker technology and genetic linkage analysis to detect QTLs for both growth and wood properties in a backcross population, a GU population, Grandis urophylla. And ultimately what we wanted to do was identify markers that we could then use as breeding tools. And this particular aspect of the study is then just to translate that QTL data into something that the breeders can use. So more user-friendly information, basically. All right, so let's talk about the population. So the population started with a Grandis and urophylla parent, which then got, let's try this. So we had that, and then we had a F1 hybrid, which is A380. So some of the tree people in the audience might know it's one of our flagship hybrids, Grandis urophylla hybrids. And then what we did was we back-crossed it to a Grandis parent, and we then had a Grandis back-cross population of seedlings. And then we also back-crossed it to the urophylla parent, and then we had both a seedling and a varietal population. So for this particular study, I'm just going to concentrate on the backcross varietal population. All right, so there was extensive phenotyping and genotyping on this population. So to start off with, if we look at the genotypic variation, so that was done with a SNP chip that had about 64,000 markers on it. So we could create a genetic fingerprint for each one of those backcross varieties. We then did phenotyping, so growth and wood properties. And what we wanted to do was just link the genotype with the phenotype to identify the QTLs, which we used in this particular aspect. But also we looked at different aspects like transcriptomes and metabolomes, and trying to see, do we find overlaps between these different aspects of the tree. Okay, so the first thing that we did was we looked at, in terms of species, where's our population sitting? So this was a principal component analysis, and we have references of, laser. Okay, so we had a reference of Eucalyptus nitens, the Eucalyptus danii, and then of course the Grandis and the urophylla. So these references were from the Genomic Diversity Atlas with the FMG group. And then this is where our Grandis urophylla backcross actually sat. So it confirmed exactly where it was supposed to be. All right, so in terms of traits, so let's start with a little bit of background on what do we find in a tree. So we find about 45% of alpha cellulose, the hemicellulose makes up about 30%, and then we get lignin of about 23%, and then some inorganics and resin. So for this study, what we looked at two different traits. So we looked at the alpha cellulose, so we phenotyped for alpha cellulose content. And this was important for us because, it has a range of uses in both the textile, pharmaceutical, plastic, and food industries. And what we wanted to do was just identify selections that had higher amounts of superior genotypes with higher alpha cellulose content than our commercial varieties, because then that has a impact in our commercial molds. And then in terms of lignin, this is quite a complex polymer, but it's an integral part of the wood. So we've seen that it's having an impact on power generation and alternative markets as well. And there's two types that are found in the wood. So you get an S-lignin, which is more easily removed, whereas the G-lignin doesn't like, it makes it more harder. So what we looked at in the trait aspect was actually a ratio of the two. So the higher the ratio, the more easier it is to remove during pulping. Okay. And then, of course, it lowers the cost of pulping for us. All right, so how did we do this? So we started off making high-density linkage maps, and we had an F2 backcross pedigree of a Grandis Urophylla population. We used the Uke chip with the 60K markers, and then we developed parental framework maps using the optimized software that's on the side. And then from those framework maps, you can then use them to identify where these important QTL regions are. And then lastly, we assessed whether we assessed the additive genetic effects and also the marker QTL linkage phase, because that made an impact whether you were actually taking the QTL across. So what did we find? So we had a total of 308 selections, right? And for alpha cellulose, we had 6 QTLs that we found, and they range from 4% to about 6% explanation of the trait. And in total, if you added up all 6 QTLs, we could explain 31% of the trait. For the SGG lignin QTLs, they range from 4 to 16%, so slightly higher. And then in total, it explained about, say, 38%. So what we did was we created a little index based on whether that QTL was present and whether and the allele present. So was it a plus, was it plus minus, or minus allele? And then also the R squared of that particular QTL. Right, so then if you just look at the linkage map, so with eucalyptus, we have 11 chromosomes, and so we found cellulose on 1, 3, 5, 7, 9, and 10, and then the S2G on 4, 6, 2 on 10, and 1 on 11 positions. Is because these two traits are completely uncorrelated. So if you look at them, there's actually nothing there. And we wanted that because then you could create a little cluster instead of a straight line. So now we could get 4 little quadrants with a, let's say, so a low quadrant and a high, and then the intermediates between that. And what we wanted to do is then take these trees, select them based on their QTL profiles. We actually went down, we chopped them out, we sent them for pulping, and now we're going to confirm whether they do fall in those quadrants. So if we look at, so the conventional breeding program, they went in also in this trial, and they went and selected just based on growth. So they did a BLUP analysis, and out of those 308 varieties, they picked 8. And if you look at it, 3 of the varieties that they picked, 3 of them were common between our proof of concept study with the 113 and what the breeders selected. But then if I went in and with my QTL selections, I actually found 2 more selections that had quite a high alpha cellulose QTL content and then an S2G QTL content. So altogether, there's actually going to be 10 selections that are now going to go into the breeding program, into our varietal site interaction trials, where it's nice little blocks, and it'll get evaluated as a possible commercial Grandis Urophylla hybrid. So those are the 2 selections at the bottom that I picked, and they were the highest SGG ranked, but yet they weren't selected by the breeders, which just went on growth. And in terms of cellulose, they actually had quite a high percentage as well. So in terms of the BLUP values, the one had quite a high BLUP value, while the other actually didn't. And yet, if you look at it, they have exactly the same percentage of cellulose QTL and of S2G QTL. But yet, in terms of their BLUP, their growth value, they actually were quite different. Okay, so if we plot this out, so the blue dots are the 113 from our proof of concept. The red dots are the 113 plus the breeding selections. The yellow dots are the breeding selections. And then the one that's ringed in red is actually the selections that I've picked. So because they both have the same percentage of alpha cellulose and SG, they both actually one on top of the other. So that's one dot, but actually one below the other. Okay, so to conclude, these are our first molecular breeding hybrid selections for SAPI, and we're quite excited. We want to test them out and see if they actually do perform as well as we think. We identified 6 alpha cellulose QTLs and 5 in total. We created an index based on the presence of the QTL plus the percentage R squared, and we now have added these QTL selections onto the breeding selection list for further testing. So lastly, I'd just like to acknowledge the breeding and the wood properties team at SAPI, the FMG group, and then the government granting agencies. Thank you. Was I quick?
Thanks, Shemaine. Any questions?
Your testing, is it non-destructive?
Yes. Yes, sorry.
Good job, Charmaine.
Thanks, Maya.
I'm actually just really curious because this is off. I understand you want to eventually fell the wood and sell it and all of that, but since you're trying to figure out how successful or how good you are at predicting, what should go into field and what shouldn't, do you have instances where what the breeders would have picked is in high contrast to what you might want as an end product consideration for your selection? And are you considering to put those in and see maybe, because I understand you're trying to weigh up the chemical, the cellulose versus maybe growth. But I think you understand what I'm asking. Okay.
So, in tree breeding, we have quite a few processes that we go through, so quite a few field testing that we do. And so usually only sort of at the last stage, at the varietal site interaction trials where you make your final selections to go pre-commercial, that is when you do your wood property testing. So there you'd find out what is its density, what is its alpha cellulose. So here we could actually make selections at a seedling stage, which is two stages before. And so the breeders do take into account, the wood property and the chemistry of the final pre-commercial selections, but they do it at a much later stage. So with the QTL stuff, we could actually do it 2 stages ahead, actually at the first stage gate of the breeding selection field trials. Yeah. Oh, sorry. You have to wait.
Hi,
Yes.
Charmaine.
Hi, Valerie.
Great work. Would you think genomic selection versus QTL? Because there's a lot of, 37% is quite high if you look at it. But from a really operational tool that you will successfully implement over time, would you bias yourself towards QTL or genomic selection? I would just, what are you thinking about that?
Okay, so I think for a little bit of background with the rest of the group here, my main job as a molecular breeder in SAPI is actually to implement genomic selection. So we have 3 different programs running, and this was just sort of a side project that, we looked at it and, there was a lot of academic work done on it in terms of the QTL analysis, and we said, well, how can we take that and actually use it operationally? But I think, genomic selection is more operationally user-friendly right now because we've put in a lot of work in there. We've got a lot of programs that's already quite far ahead. But this was just, a separate project that had been running for quite a few years in SAPI, and it was just at the end stages now is how can we translate that into actual information for SAPI itself.
Sorry, I won't ask you a difficult question. Maybe just some context. So we started this project 15 years ago, and the first thing we wanted to understand was the genetic architecture of these traits in these trees. And this is a single family, single back cross. We're systematically working from there to multi-parent crosses to large populations. So the answer about genomic selection is for sure in large breeding programs and even medium LD situations, we're going to do genomic selection, but it's perfectly valid in this single family to make use of Mendelian segregation. These are very strong QTLs, and it's perfectly valid to do the test to see whether it translates into an interesting phenotype for the company in the next generation. So just to give some perspective.
One more question?
Charmaine, the advantage of going with the QTLs is if it is polygenic, you've got a chance of catching it. Aren't you worried that if you go with genomic selection, you're going to miss those polygenic interactions?
Actually not. Genomic selection is more targeted for the more quantitative polygenic traits. So they, like if I look at genomic selection, I also use a 64K chip, and I'm targeting markers throughout the genome. So our genomic selection projects are specifically for more complex traits. That's where it's specifically aimed, and it works pretty well.
Thank you, Charmaine. Okay, thank you for a wonderful session. We will break for tea now. Thanks to all the speakers. So, Dion, when will we be back? What time?
Thanks, Chris. Thanks, everyone, for the great presentations. Okay, if we can be back after tea at 10:10. And then just, if we can please ask everyone who haven't registered for the day yet or haven't signed in for the day, if you can do so at the registration tables. And those who haven't received their gala dinner tickets, also to pick them up at the registration tables. And then for the live streaming, if you're entering through the back, please just check. We have cameras up there. Please don't walk in front of the cameras. Just duck underneath, please. Thank you.
Okay, there's still a few people joining us from the back, but our next keynote, I've got the privilege to announce him. Glendon, where are you? Can you come forward so long, please? Glendon is the lead of the research team at Stark Air, and it's a short title for actually a complex department, if you think about it. He's got about 7, please correct me if I'm wrong, 7 different breeding programs that fall under him, as well as a pathology lab, a tissue culture lab, and a DNA lab. Glendon got his PhD from UKZN, so there's some of your, yeah. So I think some of you will be glad to see him. He started his career at Pannar in Greytown. So there's also some ex-Pannar colleagues that you must be also glad to see. And then he moved to Stark Air, where he's looking after the research department now. And Glendon, the floor is yours. And sorry for starting late.
Thank you, Tonette. Morning, everyone. Just want to thank Drian and the organizing committee for inviting me here today, and it's a real privilege to be able to talk to you all and to be able to present some thoughts. I wasn't able to be here yesterday, unfortunately, so I apologize in advance if any of the points repeat a previous speaker's point, but I'm hoping to give you a little bit of an overview of vegetable breeding in the 21st century. And a bit like Zelda, I want to start off talking about a movie. She spoke about Forrest Gump. Who here has seen Wall-E? Henny, like first hand up at the back there. Wall-E. I have 3 young kids, and so I have to watch animated movies. I have no choice. But actually, before I had kids, I enjoyed watching animated movies, so it wasn't a real big problem. So for those who don't know the plot for Wall-E, it's set in the future 700 years from now, and Wall-E is the last robot on planet Earth, and his job is to clean up the planet, kind of one piece of trash at a time. And he spends his day compacting trash, and you can see these piles of trash over here. It's pretty much all he does, 700 years on. Because humans have wrecked the planet with their single-use plastics, with their pollution, with their lack of environmental control. And the humans at this point are kind of circling around space in a luxury spaceship, waiting for planet Earth to become habitable again so they can go back down and kind of colonize it. And in the 700 years, Wall-E kind of adopts a pet cockroach. And that's his only friend. And he develops a bit of a personality, which is obviously unusual for a robot. And he has a fascination with Earth's history. He collects all kinds of trinkets. But he gets quite lonely, even though he has a pet cockroach. Enter Eve, the sleek, shapely robot that he falls in love with. And she is a probe sent back to Earth on a scanning mission to detect if life is possible yet, so the humans can come back down to planet Earth. And he falls in love and ends up following her across space, ultimately to kind of rescue the humans, which is quite funny because in most apocalyptic end-time movies, the robots are trying to kill the humans, but now they're trying to rescue them, which I thought was pretty cool. Obviously, the humans by this time, being in space for 700 years, had lost all their body shape because they got lazy and didn't do any exercise. It's a great social commentary on eating habits and exercise too. And this is possibly my favorite scene from the movie, where Wall-E shows Eve his most treasured new possession, a living plant. And she's been designed to detect living plants. That's why she came back to Earth to scan and to find out if it's habitable. So her senses recognize this living plant, sets off a chain reaction which ends them up going to the mothership back in space. There's some robots who try and kill the plant, destroy it, because they don't want the humans to go back to Earth. But eventually the robots win, the humans get freed, and all is good in the end. But the scene is significant because mankind's future rests on a plant. And that's why we're here today. We love plants. We love planet Earth. And we know that ultimately our future, if it's going to be on planet Earth, is going to require plants and better plants as we approach things like climate change, etc. So the title of the talk this morning is Vegetable Breeding in the 21st Century. And this was the brief I was given by Drian. It's innovating together, plant breeding in the fourth industrial revolution, finding modern solutions to its goals and challenges. The theme of the conference is built around technology combined with collaboration to kind of achieve progress. So I had to Google, because I wasn't around hundreds of years ago, what are the industrial revolutions? So most industrial revolutions are characterized by massive advances in energy, in financial systems, in mechanization, etc., and communication. The first industrial revolution happened end of the 18th to the 19th century, where coal energy was used to develop steam energy. The steam engine, etc., produced a massive amount of mechanization. The second industrial revolution used electricity, oil, and gas, the internal combustion engine, to develop automation, or rather mass production. And things like the telegraph, the telephone, assembly lines all came into kind of being. Many say this is the most significant industrial revolution because of what was produced out of it. The third used things like nuclear power, electronics, programmable logic circuits, and robots to automate automate production rather. And the fourth one, which is kind of we're in today, and some people disagree. Some say we're still in the third, some say we're actually in the fourth. As many speakers have said, it's artificial intelligence, it's the Internet of Things, virtual reality, self-driving self-driving drones. Oh, my writing's not too bad. But let me read this. The fourth industrial revolution is growing out of the third, but is considered a new era rather than a continuation, because of the explosiveness of its development and speed and the disruptiveness of its technologies. The new age is differentiated by the speed of technological breakthroughs, the pervasiveness of scope, and the tremendous impact of new systems. It's characterized by a fusion of technologies that is blurring the lines between the physical, digital, and biological spheres, just like Zelda said. So I'd like to give you a glimpse of the future in a single slide. You ready? Plants that will tell the robot where to take them. No longer fixed to the ground. Or our new phenotyping breeders in our field. I'm kidding. Just to go back a few steps, plant breeding is a solution. It's one of several. It's a solution to feed people and increase food security. We know that population's increasing, arable land is decreasing, climate change is putting pressure on production systems around the world. And so plant breeding is a solution to feed people and stabilize economies. It's also a solution to improve farmers' livelihood. They are trying to grow crops to sustain their family, to send their kids to university, etc. Obviously, for those of us who work for corporate, it makes companies profit, but let's not talk too much about that. For the students here, plant breeding is a great solution to boredom. If you are procrastinating like I did at university, then you should take up plant breeding. It inspired me when I was in undergrad and postgrad. So it's a solution. And it achieves this by developing superior yielding varieties, either through yield or disease resistance or other novel traits, or by adding quality to those varieties. So what is the basis and how has breeding progressed through the different industrial revolutions? Breeding, as we know, depends entirely on selecting heritable traits. Those are genetically based and to some extent epigenetically based traits. It kind of only emerged as a science or discipline during the second industrial revolution, and it was propelled massively forward during the third industrial revolution. And most of the technologies, the instruments, the techniques really improved the accuracy or the speed or the efficiency of the selection process. Markers, genetic or otherwise, using simple marker-assisted selection all the way through to genomic selection, are improving our ability to select the best genotype for that environment. Things like improved phenotyping, digital phenotyping, ultraviolet, infrared, etc., drones going over fields, all of these are improving our ways to make selections because we have to select something that's heritable, otherwise we can't produce it and sell it to the farmer. So most of these technologies kind of built off of that. The fourth industrial revolution, I think, how it applies to plant breeding, has to do with artificial intelligence and data mining, predictive modeling and machine learning. Synthetic biology, I think, is a massive field which has gone forward in the last decade very strongly. and then solving problems digitally. And an example from the genomics field, pangenomics, there are much cleverer people here, I'm sure. We've started getting into this in our labs with our bioinformatics kind of crew, and you're able to predict genes and their effects using multiple sets of genome sequences to solve problems that you would never have been able to solve by growing plants in the field. And so it's a really exciting field in the vegetable side. And what the fourth IR also does is takes data from connected instruments, whether they be weather stations or wearable devices. How many of you have a smart watch? Okay, probably more than half. Mine packed up, the sensor broke, so I have a regular analog watch. If you Google, there are some wearable devices for plants. Did you know that? You didn't. So plants can have wearable devices, but taking that data from those sensors, from drones, from field probes, from robots, from phenotyping rigs, and processing that data in real time or near real time for decision making, for fine control of particular processes. Someone said a few years ago that we are drowning in data, but we're starved for knowledge. And that, I think, is the crux of the fourth IR, where data generation is skyrocketing. Every 2 years, well, in fact, in the last 24 months, the amount of data generated in the world is 90% of the total data in the world in the last 2 years. That's the amount of data we're generating. 2.5 quintillion bytes every day. And that's obviously a massive number. I think most of them are from millennials taking selfies and uploading them to Facebook. That's what I think personally. It's not actually really much new data, except, photographs, but that's maybe a personal kind of thought. But what we do with the data is critical. And this is the crux of the fourth IR. The interpreting and the application and using the data to make decisions in the context of the bigger picture. And being able to interrogate the data to make sure it's quality data. Because just because you have numbers on a spreadsheet doesn't mean the data is good. Doesn't mean that the experiment was well controlled or you had the right controls. You can't just analyze data. You have to interrogate it and then sort it out. So why do we need more data? Why do we need more ways of measuring data? Why do we need real-time information? I have some researchers say to me, Glendon, if I can't see the difference in the field, if I can't tell the difference between plant 1 and plant 2, then there's no reason for me to select it. And that's maybe a really old school approach where they like to, it's the art of breeding. You want to be able to see the difference in the plants. And that's not wrong, but I think it's incomplete. It's an old school way. It's a bit myopic in its scope. Because what happens in a breeding program is that you have the law of diminishing returns. And so as you start a breeding program, you have very quick gains. Within the first few years, 15 years, massive gains. The products that you launch are highly successful. The next ones that come out are even way better than the first releases. But as you start to mature in the program, you find a plateau where it's harder and harder to spot the difference between plots because all your material has moved forward because you've selected superior material for the last 15, 20 years. And so you kind of reach a plateau where everything looks pretty good in the field. You can't find something that looks amazingly better than the one that you released a few years ago. And so we're looking for, at this stage here, smaller differences between plants, smaller variations between plots and between varieties. And so we have to use better tools than our eyes. We've got to use things like stats. Now, when I was at university, stats was a swear word because I was in agriculture, and trying to do biometry was never fun. But we have to use things like stats. Our genotyping, our phenotyping has to become more accurate and more detailed, more precise. That's why we're doing things like genomic selection, because we're going to the extra level. Can we genotype better? Can we phenotype better, etc.? Being able to break down the components of yield and all the various kind of parts. So I wanted to maybe not get too specific, but get a little bit specific. How can an aspect of the fourth IR be applied to plant breeding? And Mr. Ashton tells us that the IoT, or the Internet of Things, is an ecosystem of objects connected to the internet, able to capture and transmit data and information with the objective of creating value for business purposes. And so, in our company at least, when we propose new technology or new implementation of software, for example, I get some very nervous breeders. And you can see when they have a bit of a nervous twitch when they come and speak to you. And they kind of worry that all the technology is going to get them out of a job. They're worried, will the genetics, will the marker-assisted selection make my job obsolete? Will everything connected to the internet, transmitting data, AI, predictive modeling, am I going to have a job? And I tell them, of course. We need someone to come in and switch on the computer every morning to make it run, right? So I've managed to allay their fears to a large part. But we do need humans. The Internet of Things has 5 components. They are the devices themselves. These are the sensors, the instruments, the probes you stick in the ground, the data loggers, etc. There's the network and its connectivity to be able to collect and collate the data in a central place. There's the data itself, and you have to think of things like how often are you going to be uploading, what is the volume that you're going to be uploading, what kind of data, and the storage requirements. There's the data processing. We've got to clean up the data to make sure that it is actually quality data that you're using. Otherwise, it's very easy to draw the wrong conclusions from a set of data where you're not sure where things might have gone wrong and messed up the sensing or the sensors and their output. And then there's the applications, the actual things you use for visualizing the data to be able to make decisions, to look at trends, to plot graphs and maps, etc. And those are all really important parts of them. But what are the benefits to that? This is what you'd need to do to get to the IoT for breeding. But I think one of the main benefits is that there's automation of a whole bunch of manual processes that can free the researcher up to do their job, which is use their brain, which is what most computer programs can't do and think out the box like a human can. And so our aim is to free up our researchers to think critically, to think differently, to think long term by automating as much as we can all the processes that lead up to that decision-making kind of process. I think also to be able to look at the data and make decisions in real time, it means you have more control over experiments. If you're going to your field once a week, you might only be able to pick up on a weekly basis if there's a problem with the irrigation, with the fertigation, with something else, with an insect. If you have data coming back every half hour, you'll be able to tell really quickly and avert a disaster where something might have happened. And so being able to control your experiments gives you that finer control. And I think also finer control means that we can dig deeper and tease apart effects of individual treatments or individual genes a lot easier. It means we can understand the biological processes underlying the phenotype, which is a combination of the genotype and the environment and the G by E, a whole lot more accurately. Because the better we can phenotype, the better we can genotype, the better we can select. The better we can select, the better the plants that we can produce at the end of the day. Just a last slide to kind of tie the things together. You'll notice that many of the startup companies in the last 10, 15 years have always been quite innovative. They've been disruptive kind of technologies, but they've always been 2 or 3 people starting a company. And innovating is critical. If we can't innovate, we should at least be an early adopter in terms of the technology or the processes or the instruments. And we know that necessity is the mother of invention, and so we shouldn't despise when we're under pressure. When the accountants say you can't have more money to do this, well, we should be glad because we'll, by necessity, invent something new or invent a more efficient process to get around the bean counters. Continuous improvement is critical. It's really important. But the light bulb was not invented from the continuous improvement of the candle. It didn't happen like that. And so we do have to have disruptive kind of innovation if we're able to do that. Breeding has its own disruptive innovation, things like GMOs in the the late 90s or the early 90s and the mid 90s and carrying on. CRISPR-Cas at the moment and the questions around the regulation of that and what that looks like for South Africa and Europe, etc. Those are examples in the breeding kind of sphere. Together, there are few areas of science where the lines aren't being blurred. When I was at university, things like tissue culture were like the really exciting big fields. And as people have gone on in those fields, they've just become tools. A tool to do something else and do something else better. And so, although there might be disruptive innovation, ultimately these new things, starting off as a field in themselves, become a tool which ends up blurring the line between kind of what we're doing. And we have to go together because although you might go faster alone, you'll go a whole lot further with others. That's how we work in teams. And so plant breeding in the 21st century, we still need to make selections. The robots can't go and do that for us as much as we would like them to, especially, who said the lazy farmers? Was it Zelda? The lazy farmers farming from their chair. We still need to trial the varieties because the effect of the environment on the genotype and its expression isn't going to change. You can't predict that phase of the process out with AI, with modeling, etc. You can't. You have to physically grow the plants, trial them, measure the yield, etc. You might find better ways to measure the yield. You might find better indirect ways of measuring the yield throughout the growing season, but you can't do away with the fact that you need field trials. There's no guarantee against nature, all this AI stuff. And ultimately, these different tools that are being developed, and we're at the start of this revolution, so the next 20, 30, 40 years, I think, will be dramatic change. But they'll ultimately lead to a much quicker process, a more accurate process for the end goal of feeding people, of making farmers having a decent livelihood and companies profit. And don't worry, students will still escape boredom through plant breeding in the future. So, thank you very much.
Thanks, Glendon. Any questions for him?
Thank you very much for your presentation. So my question is, according to your own opinion, do you think that the current education system in South Africa is aligned with these new technologies, with this AI? Are the learners prepared for this new world that we're going into?
I think there's always some schools and universities that are in line, but I'd say the majority are possibly focusing on the wrong thing. There's lots of programs around robotics and designing robots, etc., and those are good, but interpreting the data scientists and computer programmers, software engineers, being able to write software for the robot to do different things, but to be able to think about what is the biology of this process, what is the end goal, and then to be able to adapt and tell the robot or the algorithm what to do. I think we need to train our kids and our students to think critically in that line rather than building the robots themselves, if that makes sense. So I think there are some doing it well, but I think there are others that could grow as well. Dave.
Thanks, Lennon, for an interesting talk. So you gave us a very good perspective of the fourth industrial revolution, but like in your company, for example, like where you guys going to start? Or you obviously started already, but where do you put the priorities in now in a country like South Africa where you're doing vegetable breeding specifically?
Yeah, it's a good question. The tension between the exciting new technologies and some of the older school guys in our company creates an interesting kind of tension. But we've gone on the bioinformatics side, and so we've outsourced a lot of genome sequencing of our crops and our varieties and our lines, and we're looking on the data mining side because there's so much publicly available information. And gene ontologies, a gene that works in maize, if it's conserved, will work in tomatoes. And so we're trying to leverage off what's done all around the world, but then applying it to our proprietary kind of germplasm, and that's where we're starting, and that's where we've seen some quite good success so far. Yeah.
We've got time for one more question.
Is there somebody who would like to ask a question? Oh, well, Glendon, then you explained everything very nicely. Thank you.
Thanks.
Okay, Inga, are you ready?
Good morning, ladies and gentlemen. My name is Inga Gazendam. I'm from the ARC Vegetable and Ornamental Plant Institute. And thank you for this inspiring talk, but I would like to thank you for the opportunity that you give me to give this traditional scientific presentation. Sorry, just to repeat the title. So I'm going to present a single multiplex PCR that we use to genotype the cytoplasmic and nuclear male sterility genotypes of our onion hybrid parental lines at the ARC. So a very brief slide about the importance of onion. It's the third most important vegetable crop in South Africa, and it's a very popular ingredient in all African cuisine. Nobody would ever think of making a meal without starting with onion, or sheba is unknown without onion. But it's also a very healthy crop containing a source of minerals and vitamins and also some antioxidant flavonoids. So this slide is definitely not doing justice to the long history of breeding that ARC has been involved in since the early 1950s. But the breeding objectives has been to breed for long keeping ability and for multiple bulb scales. And then also in the 1980s, they started to incorporate the male sterility feature to help in the generation of hybrids. So we could also provide seeds to the requests that we received from seed companies. So very briefly, the onion cytoplasmic male sterility feature occurs in a variety of crops, and it's very basically set up by two systems in the onion crop: a nuclear component, the restorer of fertility locus called MS, and then also the cytoplasm type that can be either normal or sterile. And this is to prevent the self-pollination so that you are able to produce a hybrid. So in the hybrid breeding process, you would need a pollen donor that is able to make fertile pollen, so you use it as the male parent, and you would cross that with a male sterile female plant and then harvest your hybrid seeds from the female plant. But you also have to way off to propagate the male sterile female parent. So for that you would need the maintainer line that we also would indicate in short by calling it a B line. They are genetically identical, but they only differ from each other with the cytoplasm type. So the aim of this project was initially to incorporate molecular markers to identify the correct male sterile and maintainer onion lines, since it wasn't done before at the ARC. So this project started in 2016 already. And then as it evolved, as I used different markers to, in the end, combine the nuclear and the cytoplasmic genotyping into a single multiplex PCR to make it easier for us to apply in a large scale. So the materials and methods, you can start off with genomic DNA isolation, either from leaves or bulbs. This would depend on what stage of the season that you want to genotype your plant, so it works well from the seedlings that are sprouting in the field or in seedling trays, or you can sample from the inner scales of the bulb, which you can then still save and plant in the field after you decided that it has the correct genotype. So the CTAB method was modified a bit for incorporating the fact that it is very rich in polysaccharides and polyphenols. So briefly a slide of just the summarizing name of a few of the molecular markers that are available out there in the literature. So we didn't develop our own markers, it was the application of the current literature that is out there, which is a big field. So in the nuclear aspect, the JNerv13 PCR marker, or there's 3 SNPs that was published that are linked with the MS locus, and then the SKP multiplex PCR that was developed that is considered to be the same locus as the JNerv13 marker. And then cytoplasmic, it can be a whole variety of mitochondrial genes, or also the chloroplast, because both of that is maternally inherited. So you can use markers on any of those 2 genomes. So I tested quite a number of them. For the PCR marker JNerv, you have to separate them on a polyacrylamide gel due to the small size differences in the fragments. The SNP markers, as I said, that are linked to the MS locus, I ordered TACMan SNP genotyping assays for them. And then the ORF A500 PCR marker I could run on stretched agarose gels. When I started doing the multiplex PCR, it was a combination of the ACSKP marker and the ORF A501. And because the fragments are larger than the JNOF, you are able to separate them on a normal agarose gel if you run it long enough. Later on, with the latest application of this, I exchanged the ORF marker with the ACCD chloroplastic cytoplasmic marker and running it in the same way on an agarose gel. So all this work up until the point where I started using the multiplex assay is published in this paper. If you want to have more information on it, you can see exactly which ones we tested. And it was used to genotype 745 onion lines in 2016. I'm just going to show you a few examples of the results we obtained. So this is the page gel where the JNOF marker is separated because the small size difference between the dominant and the recessive MS alleles is very small. And this is the SNP TACMan assays. You could see that the three different ones were able to genotype the genotypes correctly, even though they separate a little bit different, but all three of them worked on the lines that we tested. And on the papers also explained how the validation was done, that it confers the pollen fertility status with the genotype. And this is an example of the stretch gel. Because it's a presence-absence marker, it generates a 473 base pair fragment only for the sterile cytoplasm. So you could run two 96-well plates on a single stretch gel, and if the band is present, you can score it as S cytoplasm. So the genotyping results in 2017, we used the nuclear TACMan assays and then the stretch gels with the ORF marker. And the results of that season, the A and the B line of one onion parental line, we could see that there was a huge contamination of the dominant MS allele, and also the cytoplasm types were not correct for the B line, the SO92. So this is when I started using the multiplex PCR assay, combining the nuclear and the cytoplasmic genotyping into one single PCR and running it onto a single gel. So with the dominant allele, they are the larger sized fragments, and then the recessive allele, the nuclear allele, is a bit smaller, and then the presence-absence of the cytoplasm type is the third band. So you'd run the agarose gels to two combs on one gel, three gels per PCR plate, and then you would score them in Excel sheets. For the size fragments that you want to make certain that you're scoring them at the correct size, I used the plant that has all three of the expected fragments as a positive control. So this is an example of how I would score and put it into a color-coded Excel sheet. And the expected genotype, if you expected it, the A line to be sterile and recessive homozygous, that was all correct. But then the B line, I detected contamination of the nuclear gene, and also the cytoplasm wasn't correct in all of the plants that I tested. So in the 2018 season, 685 onion bulbs were scored before we planted. So we wanted to discard the ones with the incorrect genotype. And again, as I say, the ORF cytoplasmic marker was used. And in this case, 4 of the lines showed the correct nuclear and cytoplasmic genotype, even though one result each of 3 of them scored as normal. But that was the part that made me uncomfortable, because a failed PCR reaction would be scored as normal because you don't detect the 473 base pair fragment. So that's when I started looking for came across another marker where the cytoplasm type is scored by a fragment each for the sterile and the normal type. So the sterile type would be 420 base pairs, but the normal would generate a band that's a little bit smaller, but not so small difference that it can't be run on the agarose gel. And then because it's combined with the nuclear genotype, you would still detect the 2 nuclear alleles. So as discussion of the results explained so far very briefly, so we were able to extract DNA successfully both from onion leaf or bulb material. We showed that the agarose gel separation method is adequate to separate the differently sized PCR fragments. And in this slide, I want to summarize the usefulness of molecular markers as I applied it. So the JNF marker was tested to validate the results, but it was never intended to be applied in a large scale because it's so tedious to run large STS-page gels. So we could do their throughput of only 30 samples over a 2-day period that includes the PCR and the staining, running and the staining of the gel. So the SNP markers, they could have a higher throughput there. If you are quick to pipette, you could do 3 runs a day. So it equates to 204 samples. But the assay mix is very expensive. So the SKP marker, that the fragments are larger, you can separate it on a normal agarose gel. Even though the throughput is lower, the assay mix that you use for the multiplex is much cheaper than the TACMAN assays. But then just before, I thought maybe it's a good idea to publish the implementation of this multiplex PCR. Some other author did, at the beginning of this year, I noticed that he already published the multiplex PCR assay, which he called the ACCN, which is for cytoplasmic and nuclear genotyping. But in that paper, he only also detected a 345 mitochondrial fragment for sterile cytoplasm. So his approach also doesn't pick up if it's a normal cytoplasm. So in the approach of the change of this approach, I think it's still more superior that the ACCD marker can detect both the normal and the sterile cytoplasm type. So as I say in the conclusion, the SKP marker, the nuclear marker, combined with the cytoplasmic marker, gives an advancement in cost and time because you only perform one reaction, so it saves on the reagents and in running the gel. And I suggest that the ACCD is preferred above the ORF 501 as a cytoplasmic marker because you don't run into the risk of scoring a failed reaction as normal cytoplasm. And then incorporating molecular markers at the ALC, we were able to quicker and cheaper and more accurately genotype the male sterility genotype of our own linear lines. And you could determine the male sterility after only 1 day after isolating the DNA. So it could enable our onion breeding program to recover the correct genotypes of our parental lines, and it could produce the correct parent seed for commercialization of our lines. And apart from the 2 co-authors of this presentation, I would like to acknowledge various technicians and assistants in the plant breeding department of ARC. Thank you very much.
Thank you, Inge. We have a few minutes for questions. Does anybody have a question?
Yes, Dave.
This is very interesting, Inge, and nice to follow up on your talk. Onion is self-incompatible, is that right? Onion is self-incompatible, and I believe that it's cross-pollinated. Because of the self-incompatibility, the seed you get is half seeds, and they are hybrid seeds, essentially. So if it's self-incompatible and you have selected self-incompatible lines and you have the right pollen parents, is it not better to go to the self-incompatibility way of doing it, or is it better to go to the male sterility system? Because the male sterility system will exclude us from using the female parents as we wish. We have to be restricted to the female parents, which will be male sterile lines. So is it not self-incompatible, and is it not good to use that system?
As far as I'm aware, it's not self-incompatible. I'm sure you could generate that in the system, but the ones we have, the parental lines, is bred to have the male sterility system. So it is you have to cross-pollinate it with a fertile donor. But if you want to develop it from scratch, that would certainly be an approach where you could avoid using male sterility, because it is tedious to bring it in the first place. So you are limited to the lines that has it in.
I think very nice gels there, but what I wanted to know is, did it work for the breeders? So in other words, did you actually do your selections at the DNA level, and then the breeders made the crosses, and did they get what they expected from your selections?
If I understand your question correctly, so we validated all the markers to confer the phenotype that we observe. Is it male sterile or not? So we definitely, with the 9 parent lines, that was the beginning of testing. Does the marker say what the authors of the markers said that they're supposed to tell you? At the moment, the genotypes are maintained, the parental lines, so no new crosses were made, but we definitely did use the genotype data to maintain the correct bulbs after discovering the contamination, to throw out the wrong ones and to only propagate the correct genotypes.
So I think it's always valuable to take that step further and show that it works at the phenotype level when you make crosses. So that would really strengthen a paper, I think, if you went through with that.
Yeah, I'll take that into consideration.
Okay, any more questions? Thanks, Inga. Okay, our next speaker are from the University of the Free State. Sorry, I just used all there in the wrong place. I'm also Afrikaans, just like Zelda. Just realized that. And it's Nutombi.
Hi, everyone. Well, malnutrition is the global challenge. But what's more alarming is the statistics that has been reported in African continent. So in 2017, the Global Nutrition reported that Africa is the only continent that faces serious malnutrition-related challenges. But they also reported 21% of malnutrition rate in South Africa, which means we are facing serious micronutrient deficiency in children. So this has led to a policy of supplementing food materials with trace elements such as iron and zinc. Now, supplementing food materials with trace element was recommended by the World Health Organization in order to meet your daily dietary levels. Now, in the world or globally, the government may address the malnutrition challenge in a short term, but in a long term, it is not sustainable. So some of you may know the reason why it is not sustainable, but I have just an obvious example. So these micronutrients are already available in stores. However, they are quite expensive, which means not everyone can actually afford them. So if not everyone can afford them, it also means they are not really accessible. So this is one of the other problems that we have. So in order to address the malnutrition challenge, we thought improving the nutritional quality of food plants, especially the micronutrient levels, will alleviate the nutrition deficiency in the world. So the purpose of my study was then to evaluate the genetic variability among the cowpea genotypes for nutritional value in order to identify the potential genotypes that can be used for the crop improvement. So just a brief background on cowpea. So a cowpea is a leafy and fruit vegetable legume crop. It is widely grown for human consumption due to its nutritional value. So its grain contains high protein and high carbohydrate and also contains a low fat. But in addition to these nutritional values that are placed on the slide, the cowpea is also known to have health-promoting attributes, such as your phytochemicals, which is phenolics and flavonoids. So some of you may know the roles of phytochemicals, which is to prevent or to reduce the chances of getting chronic diseases such as cancer, diabetes, and etc. Now, the other legumes such as your soybean and chickpeas have similar traits as the cowpea. However, what makes cowpea the most valuable crop is that it can be grown in unfavorable conditions such as your sandy soils, and also it is known to be a drought-tolerant crop. Now, this is very important if you have to consider the climate change-related challenges that the world is currently facing. So the cowpea can be grown and harvested in 5 months, and it can also be used as an intercrop. So what it means is that you don't really have to wait for your maize trial to be harvested. As long as still a warm season, then you can just plant in between the rows. So the advantage will be your nitrogen fixation, which any other legume crop have that. Now, despite the importance of the cowpea crop, in terms of the production, the size of the land where it is planted, and also the production in terms of the quantity, it is unknown in South Africa due to the fact that the cowpea producers are small-scale farmers, and they mainly rely on low-yielding and poor-performing and poor nutritional quality genotypes. So recently, ARC has preliminary selected potential cowpea genotypes for high yield and other nutritional composition. However, the information of their nutritional quality is still lacking due to the fact that the genotypes that have been evaluated are limited genotypes and also have been evaluated in limited environments. So studies on characterization and evaluation of nutritional value of diverse cowpea genotypes will aid in identification of potential parental genotypes, which will then later on will assist in developing nutritional improved cowpea cultivars. So if we can be successful in developing the nutritional improved cowpea cultivars, then we are hoping that this will contribute to alleviating nutritional deficiency in the world. So the objective of this study was to determine the genetic variability among the cowpea genotypes for nutritional value, identify genotypes superior for selected minerals, and where possible, we can give implication for the crop improvement. So we evaluated 75 genotypes for cowpea. So these genotypes were sourced from the ARC germplasm collections, and they were planted in 2 different environments. So the differences in environment, I just placed them on the table, and then we use an alpha lattice design with 3 replication for these trials. So the data was collected from a cowpea grain. I must say most of this work then was done in the laboratory, and they followed some specific procedures that are done in the laboratory to determine the minerals and the phytochemicals. Now, for the purpose of this presentation, I might just want to highlight that for each genotype, then we had 3 samples for each trait. So which means we had like 3 replications. And for the purpose of this presentation, I will only be giving a feedback or results on the first 3 minerals, which is your iron, zinc, and calcium. And I will also be talking about your total phenolics and flavonoids. So the main reason behind that, this is because these traits are linked to disease prevention, growth, and health. So the results were then analyzed on a SAS software for analysis of variance, mean values, and correlation coefficients. We also analyzed for principal component analysis on GenStat. So some of the results that are coming out from this study, we looked at ANOVA mean squares of minerals and phytochemical traits. So as you can see, we have sources of variation. So we'll firstly talk about the genotype. So the genotype effect was highly significant for almost all the traits, which indicated that there were significant differences among the genotypes. So this variation could also indicate that the genotypes, they produce different magnitudes of these minerals and also phytochemicals, and also suggesting that we could identify superior genotypes. Now, your genotype by environment interaction was highly significant for almost all the traits, except your zinc. But anyway, so the results indicated the existence of environment-specific variability among the genotypes, which also highlighted the importance of evaluating your cowpea genotypes across the environment, particularly the targeted production areas. So the R-squared values were generally higher for almost all the traits, which indicated that your statistical model represented or adequately represented the data. So the CV values were low for most of the traits, which indicated low variability in a data, and also suggesting a better way of detecting your genotypes. So as I mentioned before, we looked at 75 genotypes, and then for this presentation, we then ranked the genotype based on the mean values, and we identified the top 10 performing genotypes for minerals and phytochemicals. So this table just shows you the ranking of the genotypes in terms of the specific traits. So what you can see, the genotypes that rank number 1, it means they produce the highest mean values or concentration of the minerals and also the phytochemicals. But what we also learned from this table was that there were genotypes such as your ARC 014 and TVU 13953, which produced high mean values for almost all the traits. So I can imagine where you guys are sitting, you probably can see the balance of these genotypes in terms of the minerals and also phytochemicals. So what it means is that we can use this genotype perhaps for the crop improvement in terms of the nutritional value. So we also done correlation analysis. So the reason we did this was to determine the trait association, or to determine the association among the traits. So what we have learned from this analysis is that most of the minerals were significant positive correlated with each other, which indicated that when the selection is applied for one of these traits, then an indirect improvement of nutritional value could be observed in another trait. But what also we observed was that the phytochemicals, which is your total phenolics and flavonoids, were significantly correlated with your minerals, which indicated that selecting for a phytochemical or selecting for total phenolics and flavonoids might unintentionally reduce the content of your minerals. So we've also done a PCA analysis. So we've done this to visualize the performance of a genotype in association with their traits. So firstly, we've done a PCA, which was based on the top 10 genotypes selected based on the mean values of an ion. So what we have learned from this PCA is that a genotype such as your TVU 13953 was associated with sodium, phenolic, phosphorus, and also your flavonoid. I must just mention that also this trait shows the positive association in terms of this PCA. So what we've seen on this PCA, which was done based on the mean values of zinc, is that ARC 014 and TVU 3953 were also associated with iron, phenolic, calcium, flavonoid, and phosphorus. Again, these traits are also positive associated with each other. So what we've learned from this PCA is that these traits could be simultaneously being improved, but particularly for only these two genotypes. So just to summarize these results, there was a significant difference among the cowpea genotypes, which we said it indicated that superior genotypes could be identified. So significant genotype by environment interaction indicated that genotypes perform differently in each environment, but we also said it is very important that when evaluating for cowpea genotypes, then you evaluate them across the environments. We identified superior genotypes for more than one trait, and there was some significant positive correlation observed among most of the minerals, which indicated potential of simultaneous selection for this trait, particularly for the nutritional quality improvement. So there were negative correlation among your phytochemicals with your minerals, which indicated that selecting for total phenolics and flavonoids may unintentionally reduce the content of the minerals. So the PCA analysis was effective in associating the top-performing genotype for iron and zinc with specific traits, as we've seen from the PCA, genotypes such as the TVU 13953 and ARC 014, they were associated with flavonoids, total phenolics, and sodium and phosphorus. So these can be used to improve the nutrition, and also they have some health benefits. So the genotypes that we identified in this study can be used to develop nutritional cowpea cultivars. As I mentioned before, improving the nutritional quality of food plants, as such as your cowpea, especially the levels of micronutrients, will contribute in alleviating nutritional deficiency in the world. So we would like to acknowledge the following organizations and individuals towards their contribution on this study. Thank you.
Thank you so much. Does anybody have a question?
Nice talk, Ntombi. Are you planning on looking at the bioavailability of these minerals? You know, it's one thing to breed a variety that's high in calcium or zinc, but then if the way you prepare it and cook it, that might make some of the elements unavailable for the human to absorb in the intestine. So is that something you're considering for the future?
Yes, that's something that we are considering in future, and also including some protein analysis. So this was just the beginning of the study.
Any other questions? Thank you. Okay, our next and last speaker before the AGM is Sunette.
Good morning, ladies and gentlemen. I would like to tell you more about our sweet potato cultivar development, also from the vegetable and ornamental plant at Rudaport. I think many of you would know that sweet potato is one of the world's staple crop. It's the 7th largest crop being produced. It's particularly important for food security, but since the beta-carotene in orange flesh sweet potato is highly bioavailable, and it's an excellent source of that, it's being used for the past 2 decades to address malnutrition. In general, sweet potato contains also vitamin C, folate, some of the B vitamins, and also minerals. In South Africa, it's grown basically all over in diverse environments by both commercial farmers, growing up to 200 hectares of sweet potato, down to a large number of farmers growing it for subsistence and also as a traditional crop. But overall, the value of the local sweet potato production is estimated at 270 million rand. So at the ARC, we actually have quite a variety of aims. So at the top, you will see a blesbok, the normal purple white flesh variety. Secondly, the cream with cream flesh. So in that line, we're looking for improved taste as well as tolerance to Fusarium wilt disease. In the middle, you have the cream, which is a dry white cultivar, more aimed at the informal market, but lately we understand also that even the commercial market has a drive for that improved kind of taste in sweet potato. Then the fourth one, going to the orange flesh sweet potato, that is the Beauregard American imported variety, which is mostly grown commercially. But in that, we're really looking for excellent quality, excellent storability, looking at attractive color in it. And then lastly, the bottom one is the Popelo type. So that's a medium dry, orange and tasty variety, which is mostly used to address malnutrition. So I would like to show you some of the methods we use in the program. So firstly, a new approach in sweet potato is the use of accelerated breeding. That was started by Gruneberg. And the idea is to have fast gains. I mean, Obviously, in all breeding programs, you have a cost implication and you want to speed up the process. So instead of our normal process, we produce seed and then you will normally plant the progenies at one station and do some selections and then go out to multi-environment. This approach entails that you directly take those seedlings to variable or contrasting environments, maybe a hotspot for alternaria, drought, and some biotic challenges you might have. Then you select most of the heritable traits, like for your root shape, size, and color, resistance to diseases, and it can also include some quality traits like nutritional quality. In this way, then, you advance the lines directly into your advanced breeding stages, and you can basically release a cultivar within 4 years. So we had a population that was planted at 3 localities, Ridderplaat for high input, and also Fusarium wilt hotspot. On farm at Juzini for farmer kind of input, Alternaria hotspot, and then at Lomondo and Venda during the cool season. About 360 clones were evaluated together with some parental material. And then only the clones that was selected at the first location is considered in the other location. So it's basically a knockout type of system. And from that, only 30 lines then proceeded to our normal advanced yield trials. Secondly, we also imported some seed from our collaboration with the Southern Sweet Potato Platform, which is in Mozambique from the International Potato Center. Also, we still do polycrosses. That's the normal way that sweet potato has been developed since the 1980s. You choose around 20 or 30 parents with good traits, maybe one high dry matter, one high yield. You plant them randomized, and basically with natural pollination, you harvest the seed. Then we also use directed crosses. We introduced some material from Louisiana State University for Fusarium wilt tolerance, and then those were crossed with 7 of the South African cultivars, including Bosbok, for instance, Rabok, the normal commercial varieties, and seed was harvested from that. So firstly, those went into a glasshouse screening for Fusarium wilt. This is quite a fast method where 2 isolates were selected. Inoculum is done by using infected millet seeds, and then planting just basically 1 plant into 4 reps, and you include an uninfected control, and then use the rating scale for both the leaves and the stems. Those then, that was about 74 of the 160 did not show any symptoms, and they proceeded to the field evaluation in a natural infected field. Those were also single plant plots and 3 replicates, and basically used the same disease rating. You can see there in the middle the Fusarium wilt and also the typical browning of the vascular tissue of infected plants. 23 of those lines then proceeded into the normal preliminary yield trial. So joining from all those different approaches into the advanced yield trials, that's your more multi-location trials, at Ridderplaat, also Tompiseleka and the Lamondo station. This is the kind of data, again, the wilt tolerance in the field. Alternaria's leaf and stem blight is quite important disease as well. Of course, weevil, the normal yield data, and also then dry matter content, oxidation when you cut the roots, root defects storability just at normal ambient temperature for 14 days. Total carotenoid content, and then also your cook taste, because if the taste is not fine, no line will go anywhere. So from this, basically the results we have at the moment is that there's one variety in for release. It's called Kumo. So it's also a very nice orange variety, higher dry matter higher beta-carotene content than Bopelu, which is currently our best orange variety. And then you can also see three more lines that's out for evaluation. The one at your right, 2013-28-2, is actually a seed that came in from the Mozambican program. The other two are from the normal polycross system. With regard to purple flesh, you can see we have the two FS lines, FS 1014 and FS 1025, with a very attractive purple skin, smooth sweet potato, and also 2015-1, it's just from the normal polycross. So the FS lines are coming from those crosses with the Louisiana material, and also at the bottom one of the cream lines in that range. So just roughly, if you look at the data, for instance, the top one, 2012-34-1, quite high yield, little bit starchy line there. And then you can see the Blesbok and Ribbok as controls, one of the cream lines that's doing quite nice. What's quite important is the keeping ability. You can see, for instance, let me go to 1025, quite good storability if you compare it to Blesbok. And that's quite important if because Blesbok is also exported. Then in the orange range, very attractive dark purple skin with very high beta-carotene content, dark orange. Those lines are all coming from the Polycross system, and they are available for semi-commercial evaluation. Just a bit of data there. I think the most promising one is 2015-21-2. Very good keeping ability, and then actually the taste acceptability was very good. Also 2014-2, similar like than that. There's the Beauregard, the control, and also Bellevue. Bellevue is a new import from also from Louisiana that some of the commercial farmers are nowadays also growing, and you might have seen it in, for instance, Woolworths for sale. Yeah. So yes, to conclude, I would say that the accelerated breeding scheme is very good in terms of efficiency of the breeding, saving time, human and financial resources. Obviously, it is quite costly to take a large number of clones to various sites in your first phase, but due to the shortening of the breeding process, it's still very efficient. We do have a number of promising, we identified a promising cross-combination between the South African cultivar and Louisiana, yielding quite good purple Fusarium-tolerant lines. So that crosses, we can still repeat and try and find some more progenies from that. The lines that we get from the Mozambican program also have the advantage that they are breeding for drought tolerance. So once that we select here, we can just also test for drought tolerance and release it in that regard. I think then the various approaches enabled the development of a number of promising elite lines. So in future, I think there's a shift in sweet potato breeding that we rather go back to directed crosses, because you can you can pinpoint the parents that you use, and also your efficiency and what you get out is better. Linking that to marker-assisted breeding, which is now opened up with the availability of a reference genome for sweet potato, there is quite huge development in QTLs for various traits. And then what's also new in the market is the purple-fleshed sweet potato coming out. Of course, that's also a good source of anthocyanin or antioxidants. So there is also a niche market for that. I just want to thank our team at ARC, also the collaborators at the off-station site, and the NRF funded the Fusarium wilt resistance breeding. And with that, I thank you for your attention.
Thank you so much. Does anybody have a question? Okay. I'm the one in my household doing the cooking, so I just want to find out from you for the taste of the different varieties. How do you screen for that?
Yes, so in the early phases, we just do raw. You have to go and cut and taste, well, 100 or whatever lines. Then in the later phases, we physically cook it, and we use small taste panels at the, at the institute or off-station sites.
Sunette, when you do your sequential knockout, obviously the sequence of the locations is very important.
Yes.
How do you go about selecting those?
Yeah. So sometimes you are more forced because of the climate. For instance, at Roodeplaat, you can only plant at a certain time, but like at Venda, you can go into the cooler season. So yes, I think we didn't really focus on what you're saying. However, if I found a line that was really doing very well at maybe the later locality, I did go back and check if it at least was a medium well yielder, and then I kept it in. So yes, it was mostly a knockout system, but not deadly.
I see in that cohort that you evaluated, there's 2014 and 2015 lines. Do we all have the same problem that you get behind with some of the lines in the screening process?
Yes, and also if you don't have quite a number of, if you only have 10 lines, it's sometimes not a good trial to just do the 10. So I tend to then rather combine them. Maybe they're a little bit earlier phase, but rather combine them that you can compare them properly with each other. But then, of course, in the advanced yield trial, it's possible that different years' material can be evaluated.
Thank you. Any more questions? Okay, then I'm going to hand over to Drian for the AGM. That is a closed session. Drian will explain all of that. So while he is setting everything up, we just want to ask the Stark Ash team for a group photo with Glendon before he disappears. I think maybe if we can just all meet outside, and then it will be quick. Thanks.
Thanks, Tonette, and thanks for all the presenters during the morning. For the live viewers, we'll be back at 1 p.m. And then we'll move to the closed session AGM right now. Everyone who's not a member of the society, you're allowed to leave. Thank you.
Hello, can everybody please take their seats?
At Corteva Agriscience, we're developing sustainable, integrated, whole farm solutions that are reinventing the world's oldest industry as one of the most modern.
Okay, good morning everybody and welcome to the Corteva student organized session. Firstly, Drian will give us a brief overview of the history of the Plant Science Symposia with Corteva.
Thanks, Ingrid. So you guys might have seen the hashtag at the end, keep growing. So these nice little plants look very cool. I hope they keep growing. So just some background on the Plant Science Symposia series. So the whole idea around the Plant Science Symposia series, which started in 2008, is to engage students in an environment and support the plant scientists of the future. So it's to create a platform of learning, networking, career development, soft skills. It's really for students. That's why we have a student committee who's really organized this whole event from actually the live streaming we've been using all day to these plants we have here. And there's a tremendous amount of work in which they've helped, not just for this event, but also for the SAPBA Conference. So it's a global platform, and we've had plant science symposias throughout the world. And we've had partnerships at Corteva with organizations like the SAPBA across the world to host these mini symposia within the bigger conferences. So it's really about collaboration, networking, keeping us talking about plants and how do we improve plants. So, yeah, just some of the stats. So up to now, it's more than 150 events which have been hosted by more than 60 institutions from 16 countries on 6 continents and over 30,000 participants. And I think those 30,000 participants exclude the live streaming participants we've had. So we reach a real broad audience to talk about the plants. Just some of the connections and connecting students across the world and the international network of speakers. So for this afternoon session, in the later session, we're going to have Dr. Emerson Limburger phone in from the US. He unfortunately couldn't be here because of the coronavirus. But thankfully, we have the technology to enable us to still bring the message across and to connect. So there's a Plant Science Symposia series web page from which anyone can view these live streaming events. So as you'll see, there's many events happening almost every other week across the world. Go visit the web page, dial into the events, be part of the conversations in the academic space. Encourage your students to go and connect on these platforms. And that's it from my side. I just thought I'd give the brief introduction. What is the Plant Science Symposia series? I didn't want to hog too much space from the student organizing committee. And I just want to say to you guys, job well done. I think it's going to be excellent. Thanks, Ingrid.
Okay, I would firstly like to introduce the rest of the students that helped to make this session possible, so if they can just come down quickly. Okay, so this is the committee that was chosen. So we are all from the University of Pretoria. So we have Anna Mshlangu, Tanya Welgemoot, Tristan Nadasen and Robert Janssen van Vuren. So yes, we all organize the little plants you see in front of you. It's little spatbooms, so if they root, you're welcome to plant them in your garden. And yeah, we hope you enjoy our session. Thank you. Okay, so our first keynote speaker will be Prof. Mark Lang from the University of KwaZulu-Natal. He is the director of African Center for Crop Improvement. He is also a formidable academic who have helped graduate 84 PhDs and 86 master's students. So who better to give a quick overview on how to manage the timeline of your thesis. Thank you.
Thank you. Thank you very much. This is a bit crazy. I should never give my secrets away. Getting a PhD is a highly stressful process, and I've had success and I've also had delays. And I think the most delayed PhD that I've worked with is Nigel Gretsch, who took 25 years to get his PhD. That was inherited. Okay, the ACCI is just a specialist unit at UKZN, and we've been running since 2002 with funding from Rockefeller and then subsequently the Gates Foundation. And our mission was to train PhD-level African plant breeders on African crops in Africa. And the reason for that was because Rockefeller had tried previously to train African scientists in America, and 2/3 didn't come back to Africa. So it is an enormous brain drain and waste of money. So we've worked in 16 countries, 20 crops, and with 100% retention in Africa, which is pretty nice. And all of those people have remained in agriculture, mostly in plant breeding, sometimes in the seed system, some at universities, but largely in the plant breeding business. And 123 graduates so far. Unfortunately, we don't have Gates funding in future for, they've stopped funding education. They say they've done enough, which is a really weird concept. The challenge we faced, and it's one that I guess a lot of you face if you have NRF bursaries, and that is that we had a fixed budget and each student had to graduate on time. We were lucky enough to get a 4-year cycle for each student, so 1 year of academic training with us, and then 3 years of field research in their home country at their home research station, with the idea that the supervisor would visit the student rather than the other way around. So the problem we faced was that for political reasons and financial reasons, we simply couldn't afford our students not to pass on time. They had to submit on time and they had to pass. So we had to drive the process. It couldn't be a passive one, which is the old model of universities. You get an idea, you knock it around, you chat to your supervisor every 6 months. That simply wasn't going to work for us. It had to be a much, much more controlled process. If we look at the global situation, Australia, about 65% completed their PhDs in 4.5 to 5.5 years, instead of the 3 to 4 years that they were meant to complete. So running a year or two late. The UK, 57% completed after 5 years, and then 71 after 7 years. 7 years, and it's meant to be a 3-year PhD. So clearly there's a problem there. And part-time students had a very poor prognosis. I mean, 19 and 34% completing. It's not great. US, it's about 40 to 50%, but that's after 6 to 7 years. Okay, they do a year of pure academic studies, but then after that it's fieldwork, it's studies, it's research. So you're really looking at 5 to 6 years of fieldwork before they submit. Canada again, pretty slow, And UKZN, it's about 5 years, is for about 75% to graduate. As I said, the one case was 25 years. My own case was not quite as long, but it was far too long. Dead slide. Okay. This is someone else's help. Okay, what are the consequences of PhD attrition and slow time to degree? Well, it's stress for the PhD student. And there's a recent paper, a Nature survey, showing that 36% of students were anxious or depressed to the extent that they needed assistance. And a UK survey again this year, 86% of the students reported severe anxiety. So I think that goes for the postgrads and for the supervisors. So when PhD students drop out or are excluded, it's a lose-lose situation for everyone. And the student loses, the supervisor, everyone loses, the funders lose. It's a disaster. And ironically, there's little difference between the intelligence and prior academic performance of students who drop out, because it's generally not about that. It's really about organization and psychological issues and the management side of a PhD. So unfortunately, PhD candidates have become a disposable, replaceable unit. Any disposable, replaceable units here? So it's crazy to waste it. So our strategy for managing it was to try and manage each PhD student as a project on their own. They are a project. And their thesis is the project that we have to focus on. And the first point I make there is avoid writing. One of the most common mistakes is that people leap into writing. And particularly the more diligent, hardworking student, the more likely they are to sit down and write a 50-page dense lump of indigestible ideas, which doesn't help at all. And in fact, the whole trick is don't write. Capture the core concepts as concepts and make them transparent. And the trick to do that is to use mind maps. And mind mapping is an incredibly powerful tool for capturing concepts and ideas. And it avoids all the writing, and it avoids hiding, because the moment you start writing, you can hide. And so people who are scared or insecure or not sure of what they're doing will write, and they will hide in the writing. So for supervisors, again, please use this powerful tool, because it allows you to insist on transparency. And the student who doesn't understand is exposed immediately, because it's there, it's obvious, it's a picture, and pictures are visual and immediately available. So it's an incredibly powerful tool for that regard. So from the mind map, then it's really easy to convert it into a project plan. And the best way to do that for plant breeding, and it's not always true for other subjects, but pretty much, you can create a flow chart. And the advantage of plant breeding is that it's always seasonal based, and it's always a cyclic process. So it really lends itself to a flow chart. And the advantage of that is that a lot of project planning and the whole science of project planning, and also the software of project planning, is available essentially as a flow chart. So we look at project management of building a building, it's all a flow chart. And it's looking at the resources, it's looking at the log jams, it's looking at the budgets and the timescales of all of it. The timeline, you can have it as a very simple timeline. And just a seasonally based timeline is so powerful when you're doing plant breeding studies. But the thing that most international funders want is a Gantt chart. So it's not a complicated thing to do. it can get as complicated as you want it to do, and there's lots and lots of YouTubes on how to do a Gantt chart. But the bottom line is you need a timescale. And that's been the real trick, because if you know what the timescale is, then as PhD students, you know what the end point is. And one of the single biggest causes of students not finishing on time is they don't know where the end point is. And they don't have a deadline as a time, as the 1st of December 2023. That is your deadline. You have to finish by then. There will be no compromise. And knowing that deadline changes everything. And I use the analogy of comrades. People run the comrades, and you watch them running, and they come into that stadium and they run round, and then they get over the finish line and they collapse. And I always wonder, why do you collapse? And I think the answer is very simple. They can push themselves to the finishing line. And we need to give finishing lines to our PhD students. And that is the finishing line, and there's no compromise. And you run to that finishing point. The resources and financial budgets are crucial as well, because it's really tragic when you have a student who wants to do something and it's way out of budget. You know, they want to go and use an iron torrent machine, and it's a small project. It's not going to happen. And that kind of misunderstanding needs to be resolved right at the beginning. If you get lucky and if you find funders, yes, maybe then things can change. But right at the beginning, there needs to be a clear understanding of what the technologies are, what the costs are, what the timescales are of all of those. So it's about the clarity of thinking and understanding right at the beginning. Once that is done, it's actually possible at that stage to create a contents page. Now, it's a rough draft of the contents page, but it creates an intellectual framework. It's the intellectual skeleton that drives the entire PhD. Will it change? Yes, it will. Many of those chapters won't stay the same as they are. They have to change. It will be dynamic. Things won't work as expected. It doesn't matter. What matters is that there's a plan and there's a map and there is a pathway. Most importantly, there is an endpoint. And it's having that clarity of endpoint and a path to get there. That's the crucial part. Adaptation on the way, changing things because things don't work as you expected, that's normal. I think that where industry funds, they always want reports. And again, that's crucial to put in so that it's transparent right from the beginning. When are those reports due? And the interesting one is the progress meetings. Some people are meticulous about them. They have them weekly. Some people have them monthly. And then some people say, come and see me whenever you want to. And I think that's dangerous. And really having formally scheduled meetings is, I really believe, is crucial. We spent a lot of time on enabling skills. We spent a year on enabling skills with the ACCI. And that included writing the project proposal after the mind map, etc., and also an extended literature review. But there were a range of other skills that we taught, including the mind mapping, the project management and budgeting. We actually used MS Project as a tool, which we taught specifically to the students. The oral and poster presentation skills are interesting because they make a profound difference to the confidence of the students in a public forum. And one of the interesting things is that we had funding from Rockefeller and then Gates, and actually Melinda met our students a number of times in the field, and the thing that blew her away was the self-confidence of our students talking about their crops and what they saw. The skill sets for attending local, national, and international conferences is an interesting thing. People assume that, well, you just go to a conference and you pitch up and you go to it. And I'd like to believe that there are actually a set of skills that you can teach about how do you go to a conference and how do you get the most out of it. The first big international plant pathology congress I went to, I tried to read all 2,000 of the posters. And I blew my mind. I literally blew my mind. I mean, I just had a loop in my head of all these posters. That was just naivety and ignorance. I think that kind of thing can be taught. And I think asking the right questions and being engaged in lectures, whether or not it's at a national or an international level, I think that PhD students are in a position to ask questions about their speciality area, and they should be asking those questions. So how do they frame them? How do they get those questions asked? And it's possible to have an impact even as a PhD student simply by the quality of the questions you ask. Scientific English is an interesting challenge because it has utterly different rules to humanities English. And we know this because we've tried using humanities English teachers, university level English teachers, and it's been a dismal failure. And they just didn't get scientific English. And it's a different level, and most ordinary human beings don't get scientific English or Russian or Chinese, because as scientists we write in a very formal way, and it's teaching that's crucial. Yes, there's the fundamentals of English underlying it, but it's, as scientists, how do we differ from ordinary writing? I think plagiarism is a problem that we all face. I don't know how the other academics feel about it, but for us it's a huge problem, because there's more. It's very easy to do plagiarism, and yes, you can use things like Turnitin to test it, but the reality is, what is plagiarism in a world of texts online which are easy to cut and paste? And how do we manage that? And a lot of our students really didn't understand how serious it was, and that it was actually an issue. And some had grown up using that as a standard way of writing articles. So there's a serious area that needs to be engaged with and discussed and debated and resolved. The obvious ones of accessing the literature and referencing, but the reference management is particularly interesting because there are software tools like Mendeley and EndNote. The problem with them is that they're flawed. Okay, one thinks that something like EndNote is good and it works. No, it doesn't. It's actually a very flawed piece of software, and what you put in doesn't always come out as it should. And if you change from one journal to another, you hit the right button, and it still comes out flawed. And so ultimately, you actually have to do everything manually and check. You might use it as your database, but in fact, you've got to actually check each and every reference, because rubbish in, rubbish out is the first problem. The second is that the software itself sometimes has flaws and puts a colon where it should be a semicolon, and you have to fix that. The advanced biometry skills are interesting because one of the challenges we have is our students come from other countries, and in most of those countries, they don't have the money to buy licenses to SAS or GenStat, and that's a real problem. And so I think we are moving much more to R and the use of R in the long term. Because it's an open source software package, I would suggest that that probably has to be the future, and that GenStat and SAS need to be running scared. UKZN nearly pulled the plug on both of them last year. So to me, it's just a question of time when the plug gets pulled on them. Participatory rural assessment is something that we insisted every one of our students did, and that was to meet something that Shimilis is very fond of, which is to meet the farmer-led plant breeding concept. And so teaching those skills is absolutely crucial, and it's not something that plant breeders do naturally, because it's a whole range of soft skills: questionnaires, interviews, group meetings, and then the software to manage it tends to be SPSS, because the data is all non-parametric. So you need to work with a different set of statistical tools. But you need those statistical tools so that the surveys you do actually have validity and are publishable. Fundraising is something that doesn't appear necessary for a PhD student, but in fact, if you can involve your student in the fundraising aspects of engaging with funders, it's brilliant. But in the long term, their survival as a scientist and their success as a scientist will come down to how much fundraising they can do. And I think that goes for all of us. At every university, we're all trapped in that paradigm. And I think that in the ARC, you've got the same one. And across Africa, you've got the same. At most of the national programs, unless there's external funding, you're not going to be able to do research. Publication skills are crucial. But the interesting one was creating a culture of publishing. And when we started out at the ACCI, I have to admit that we were very poor. And we didn't publish much out of our students' PhDs, and we hadn't created that culture. And it's something that we've worked on, and it's now very interesting that it's a real drive. So to us, it appears as a culture. It's not something that happens overnight, but it is a culture that you shall publish as much as you can out of your thesis as soon as it's ready. And that's the other thing. Writing up at the end in the old-fashioned way as a big book is an old model. It's an archaic model. And it doesn't actually fit in a world that's moving very fast, because you actually need to publish your research as soon as it's done. And so each discrete chunk of research that gets done needs to get published as soon as possible, because you face the dilemma that someone else might publish it anyway. And then finally, the focus on the PhD completion with no safety net for delays. That's just a fundamental concept that we all need to work with. The delays that do happen, the number one cause I'd like to suggest is the fear of being judged. So I found that the people who have had the greatest problem of writing up end up being perfectionists. Perfectionism actually is an escape from being judged, because you never hand anything in because it's not perfect. And if you don't hand it in, you can't be judged. So as for the supervisors here, watch for that, because it's a very, very powerful tool. If you recognize it, you can deal with it. If you don't recognize it, you'll just see that student as being a perfectionist. It's actually a problem, and they probably had, if you want to go into the pop psychology of it, you'll find that either of their parents was very demanding and very critical. And it's every time a coconut. Okay, writing skills are really interesting. Tenses are a nightmare. I've studied Italian and French, and as Nikki will know, my Italian is terrible. And knowing the tenses is really hard. The other one that's interesting is articles, and the. And again, Italian is interesting because it doesn't have articles. But most African languages don't have articles either. And I thought that it was pretty easy. But we went to the Cornell website on language, and there was 15 pages on articles. So it ain't easy. And then punctuation is a challenge. The paralysis that I also discovered in writing up is in the discussion section. And the introduction, materials and methods and results are basically reporting a history, so it's quite easy to do. Occasionally, a student will get stuck on a statistics analysis and what it means. But generally, it's when students come to writing up the discussion that they get paralyzed. And the reason is that the discussion requires judgment, and it requires comparing and contrasting, and also very often dealing with conflicting results. And that's hard. And it also requires exposure, because now you have to write down what you think it is, and that exposes you and your thinking and your understanding. So it's the time to feel very exposed. So our solution was to deconstruct that process and break it down into, number one, capturing the critical points in the results, just in point form. No discussion, non-judgmental, no missing items. No missing items means that there's no worry at the end of writing the discussion that you need to go back and look again. Did I miss something? It's done. And then writing a paragraph on each of those points. And then the third phase is to mix, merge, and smooth those paragraphs, combining them, putting the appropriate things together, and then having the linking phrases like, this was found, blah, blah, blah. However, this was a contradictory result that we found, and it was also found by Jones and Brown. So that's the stage when the matching and mixing of both the students' own work and also external parties comes into play. And this seems very simple, but it's incredibly powerful, and I hope you'll try it. The discussion is where people get stuck, okay? And this is a way to unravel it. And the fear disappears, and the paralysis dissolves, and that's a miracle. So I do think that we can manage the unmanageable. The initial planning is crucial. The endpoint is essential. Teaching those enabling skills, I don't know how you're going to do it in your particular circumstance, but having it on a block basis is brilliant because you can have short, intense blocks with visiting lecturers. You can have a 2-day workshop, which is 8 hours a day, and you make it really intense, and then people really gain something out of it. The project plan is there as a template, and it's really great because it's got a timetable, it's got a budget, and there's a progress plan. And it's really great for the student and the supervisor to be working with that progress plan. And then the writing of, please manage that perfectionism and the discussion section. So we need fast, flexible, innovative breeders. And here's a way to get our breeders into the professional world on time. Here's Joseph Kumar. He's a cassava breeder in Kenya. Cassava is notably slow, and it's difficult. And I think that plant breeding is one of the toughest subjects to do and finish on time. But he managed to get his PhD completed with 3 years of field work. Okay, he got his original parental crosses done in the 4th year, but it was actually 3 years of field work. And that's a very slow crop to work with. And then Jeffrey Kananji managed it again with 3 years of field work. And both of them ended up releasing varieties. So it is technically feasible. We've done it. We've done it with slow crops where we haven't been able to hurry. It's not a case of working harder. I always envy the biochemists because if something goes wrong, you can redo it, and you can redo it in a week. You just have to work harder. You have to work overnight. You can't make a cassava plant work harder. So we have graduated 83% on time with 12% a year late, usually because of drought, and we've had about 5% dropping out. So that's way better than what the university norm is, particularly at getting it on time. Thank you very much.
Okay, thank you, Prof. Lang. Do we have any questions for Prof. Lang? Yes.
Thank you, Prof, for a nice presentation. I have about 2 questions. One is, how does one, given the different background that we are all from, how does one find the balance between having to take care at home and studying for PhD? How do you go around that one? That's the first question. The second question is student-supervisor relationship. At times you find that it's not so right, and then it tends to hold back students from graduating. So how do you then go around those things?
Okay, sorry, I don't understand the first one. I mean, I think that there's a huge problem for many students who come from a poor background.
Yes, yes.
And money is an issue.
Yes, yes.
And so I would say that the fundamental of all science is this. If you don't have money, you can't do that research. And so the fundamental for a PhD is if you don't have a bursary, it's going to be really, really difficult. And there needs to be the money to meet that budget. So that's why I spent a bit of time talking about the budget. And I think that the bursary is part of that budget. And it's really bad. It's a bad idea to start a PhD if you don't have a clearly defined budget for the bursary and a clearly defined budget for the research that needs to be undertaken. Because you're just putting enormous stress on something that's already very stressful. A psychologist friend of mine who did his PhD at the same time of me said that neither you nor your spouse are ever the same again after you've done a PhD. I don't know how that rings true for all of you.
But then what I'm saying is that if I have a very poor background, I should just neglect working and just continue with PhD.
No.
And let them suffer for a while.
No, you've got to go and find that bursary, and you've got to find a supervisor with a funded project that can fund that PhD properly. And if you spend time finding that bursary and spend time finding a supervisor with a really good fund for a good project, then you'll succeed. Your second question is about supervisors, and that's absolutely right. The relationship between a PhD student and a supervisor is critical. One of the ways to try and deal with it is to have at least two, but then I had two supervisors and they didn't like each other and they fought, and neither of them would talk to me because they were fighting over me and what I was doing. So I was the ping pong ball in the middle and it was awful. So that doesn't always work either. So it's a thing of trust, and you have to find a supervisor that you trust. And you also have to have supervisors that trust you. And are you trustworthy? Do you plagiarize? Can you be trusted with the funding? Can you be trusted with an expensive project? One of my colleagues was doing research on platinum, and her student poured the platinum down the sink. It was a very expensive mistake. So yeah, it's hugely challenging. So get that right. Find the right supervisor.
Thank you. We have time for one more question.
Prof, it's a, can you hear me? Okay.
Yeah.
It's a very, I think, interesting subject that you touched on, particularly on students and everybody wants to continue studying. My question is based on the mind mapping, basically management as a whole, as you want to put PhD as a project on its own, and you've got your supervisor who has to manage that project and the student managing their own project as a PhD.
Yep.
Mind mapping, I believe, is a skill that you build over time. It's not something easy. I had an opportunity in my past to learn it in undergrad, particular subject. So what I'm asking is at what level do you introduce to students that mind mapping? Is it boom on PhD level? I don't think that 3 years when you have developed a skill of mind mapping, you're gonna get all the, On your opinion, at what level do you, as a professor, do you think that mind mapping teaching should be introduced into
I think that's a very important question, and I think that it's such a powerful tool that it should be introduced into undergrad courses, so that certainly at an undergrad or possibly at the honors level, we're teaching it. We teach people statistics because that's an essential tool. I really believe we need to teach mind mapping and project planning at the honors level, which is really where we're preparing students for a career in science. So for me, that would be the ideal place. The challenge that I think we have, particularly with the ACCI, where most of our students were from other countries, we didn't have that opportunity. But many of the students that we take as postgrads weren't there as undergrads in our undergrad courses. So they're arriving with a different set of skills. So it's often, that's the first crash course that we do for PhD students or master's students when they arrive and start working with us. And it can be with them personally, and it's working with a supervisor who is experienced with mind mapping, and they do it together. And it's something that can grow and evolve as opposed to just arriving with the finished product. So yeah, absolutely. It needs to evolve and grow. It's an incredibly powerful tool, and you never stop learning how to do it better.
Okay. Thank you, Prof. Lang. Thank you for sharing your secrets with us. All of our students could benefit from it. Okay. So our next speaker is Maya Mostert-O'Neill, and she will be talking about domesticating eucalyptus for changing climates.
Thank you. Good afternoon. I am very loud. I've never had a microphone like this. I feel like Beyonce. I am a PhD student, and I'm 20 days from going overtime, so thank you for that pep talk. And I work in the Forest Molecular Genetics Research Group under the supervision of Professor Zander Meiberg. So this is work that I did towards the PhD. As Joel introduced yesterday, Eucalyptus grandis has been in South Africa for over 100 years. Initially, South African foresters liked it because it grows fast, it had excellent properties, and it was used for solid wood timber in the mining industry. Later, it also became an important species for paper and pulp production. Of course, breeders had to navigate, among other things, like the end product that they wanted, pests and diseases, and we know that the challenges are heating up for them. We'll circle back to this issue at the end of the talk. So a lot of the marker-assisted breeding that you saw so far in the conference, and that you'll probably see also tomorrow, involves having a trait of interest, so maybe a disease resistance, specific chemical properties, or an end product, and then finding the genomic regions underlying that trait so that you can find candidate genes or candidate markers with which you can select. But what I'm going to show today is a completely different approach. It starts with the genome. And without having any trait in mind, we're just asking which regions of the genome show signatures of selection. These signatures work as such. If we look across the population, you would expect as the population grows, as we change things, the genome, all the markers in the genome will change by more or less the same frequency. But if there's selection pressure on a specific part of the genome, then that region will have a different inheritance pattern from the rest of the genome. And we can detect those patterns as signatures of selection. If we know which genes are in linkage with those markers, we can begin to understand which processes have we been selecting, be it intentional or unintentional. And I highlight here seedling development, below ground properties, and even environmental interactions that we are not consciously selecting. So this was the premise from which we approached this project. And the question we were asking is, how has nearly a century of artificial selection in an exotic environment changed the genome of Eucalyptus grandis? Specifically, which genomic regions have we been targeting? Now, of course, to answer that, we first have to know where did it come from. What was the genetic diversity already present in the wild that was brought to South Africa and then cultivated here? For this, we imported seed from the natural range in Australia. All the seedlings we germinated is now established in a gene conservation park in the Natal Midlands. This was done by Mondi. And in total, we have representation from 33 provenances. And these were seed collected from 194 wild-growing seed parent trees. We SNP genotyped using the SNP chip, almost 600 individuals, and then we subset about 360 individuals so that each family is only represented by 2 half-siblings. So we don't have overrepresentation of a particular family. For everything I show today, the genotyping is with a Eucalypt SNP chip. We get about 20 to 25,000 informative markers off that chip for Eucalyptus grandis. So initial population structure analysis of the wild material, we saw that the Mackay provenances there in the middle of the distribution seem to have differentiation patterns from the other subpopulation that was a little bit more extreme than what we usually expect for within species differentiation for eucalypts. And this led us to question whether there was interspecific integration in those provenances. To test this, we modified an approach called ancestry mapping. So with ancestry mapping, you take each region in the genome and you say, what is the most likely ancestral source, given a set of reference populations? Now, we took published SNP genotypic data for different species. The first is, of course, we have our own Eucalyptus grandis. That was going to be reference population, ancestral population 1. And then we have different species from the same section. So the section of Lotta ungulata. So these are non-grandis Lotta ungulata, same section as grandis. And then we also had 3 species from a completely different section, the Maidenaria. And what we did is, for each marker in the genome, so here we have one individual and the 11 chromosomes of Eucalyptus along the x-axis, and for each marker, which is a little dot, but there's so many, they look like lines, we could say, is that marker, is the one allele from, are both alleles from grandis, or is one allele from another source? And the example I have here, chromosome 1, have all the markers on chromosome 1 assigned as one allele from grandis and one allele from a different species in the Lotta ungulata. Of course, we had to check whether this works before we can chuck it in to test all our samples. So we analyzed, did ancestry mapping of individuals where we know the species should be pure. So we have 2 examples there at the top of pure grandis, so all the markers along the genome assign as both alleles coming from grandis. And then we also have examples of a Lotta ungulata and 2 Maidenaria. We can look at hybrids. So F1 hybrids will have ancestry between grandis and the other species with which it was crossed. We have a backcross individual over there and at the bottom. And we even looked at 3-way hybrid cross, so Grandis europhila hybrid crossed with a Grandis nitens hybrid. And you can see how the ancestry switches. And these are actually the recombination events during the formation of gametes in those parents of these hybrids. So it worked. I was very chuffed. And when we did ancestry mapping for all 600 individuals that we SNP genotyped, and for those in Mackay, there were over 40 individuals. I'm just going to show a little subset of them. We could see that all the individuals had extensive ancestry assigned to something other than grandis. Most of the time, it's one allele grandis, one allele the other integrating species. So we don't know what the species is, not for sure, but we know which regions of the genome is affected, and we know that there is integration. Next, we wanted to see which environmental factors explain the integrace population structure, and aridity came up as a very strong predictor, environmental predictor. And we know that 5,000 years ago, this part of Australia was quite wet. It was the perfect conditions for Eucalyptus grandis. And then since then, the El Niño Southern Oscillation weather system came into play, and a dry strip started to develop right across this channel where the Mackay provinces are. So we believe we have found evidence of adaptive integration in response to increased aridity. So the next question was, how much of that wild genetic variation do we have in South Africa, and how does it relate to each other? For this, we got material from advanced breeding populations from 3 companies. And just to remind you, I think Joel mentioned this yesterday also, Eucalyptus grandis was part of a government breeding program first, and then the populations from SAFRI, some of them were taken up, different genotypes were taken up by the 3 companies and then advanced in private breeding endeavors. We had over 800 breeding samples from the 3 companies. And when I looked at the population structure of these breeding companies in relation to the wild and to the other Lotta ungulata species, we saw that along the first component is where the most variation is, so that's Europhila, that's all the grandis. The second component separated the wild from the core breeding grandis, and we can see a lot of our breeding samples appear to have interspecific integration. And we suspect it is from Europhila because GU hybrids are grown close to these populations. We also see evidence of recent genetic infusion from wild material. And as Joel said yesterday, that is common practice. To keep the genetic diversity up, they infuse from unimproved or from other breeding programs. So we can see all this evidence, but interspecific integration at a genome scale, if you're looking for differentiated regions, those regions will, if you have interspecific integration, it kind of, it just stands out. So it doesn't answer your question about artificial selection. All you know is it's a different species genetic material that's in there. So we had to exclude those. And then, I can't go into the details, but we also excluded recent genetic infusions because that will dilute that signal of what we've been selecting for over 100 years. In the end, we have clear distinction between the breeding and the wild material. So here I have the southern subpopulation of the wild, the northern subpopulation of the wild, the breeding material. We ended up with over 500 individuals for the breeding. And I left out those two Mackay provenances because we don't see any genetic or historic records to suggest that the Mackay provenances were ever introduced into South Africa. So we can't expect that, that it was differentiated from there. Okay, so this brings me to the last question, or the last objective, which was to identify genomic regions that's differentiated between the wild and breeding populations. The first method is a very crude method to identify what we call outliers. So those are genomic regions that have inheritance patterns that is distinguishable from the genomic surrounds. And how PC-Adapt, which is the tool I show here, works, is that it looks along, in these samples, along the first PC, and it looks at their genomes and looks for markers that are differentiated along that axis. And along that axis, those ones that are differentiated there are colored in dark blue. You can see a massive peak there, and then some others scattered around. And then the next level is those that are differentiated along that axis, so they are in turquoise. Now, with PC-Adapt, it doesn't see groups. It just looks along that axis. So it can even identify regions that are differentiated along this axis within a cluster. The next method, using discriminant analysis of principal components, here we define the groups, and luckily it does split very nicely into the wild individuals and the breeding. And here we're asking which markers are best at separating wild and breeding from each other. So those are called SNP loadings. In other words, a high SNP loading means it has a lot of information for you to separate these two clusters. And again, using that, we pick up on chromosome 4. Remember, it's the chromosomes along the axis here, that big peak that we saw before using PC-Adapt. And of course, the last method is marker-specific FSTs. FST is normal differentiation measure, and it's probably the most finicky, so you really only pick up those that you're pretty sure about. Okay, so that region on chromosome 4 is enormous. It's quite unusual. And if we look a little bit closer, here I just zoomed on that chromosome. If we look at linkage disequilibrium across the breeding and the wild populations, we see that linkage disequilibrium is very low. That's because it's an outcrossing species. But we do see a little bit of a higher LD cluster over there. And if we zoom in on that area specifically, we can see that there's actually quite significant LD there. And more than likely, something in that area where that one outlier is very clearly, somewhere there is something that is actively selected. And these other markers are just hitchhiking along in the genome because they are in linkage disequilibrium. And because we haven't been breeding or putting this artificial selection pressure on this area for many generations, this big block of LD hasn't had a chance to break down through recombination into a smaller bit. But this is not a region of high LD in the wild. It is really specific to the breeding. So we believe that this could be a selective sweep that is present in the 3 breeding populations. It's a pretty nice finding. Well, anyway, hopefully the students like it. Anyway, so to conclude, we were able to identify genome regions that have signatures of selection, and this would be in response to artificial selection. Next, we're going to look at the genes that are in linkage with these loci, and hopefully figure out something. Maybe there's a functional enrichment for those genes that can lead us into a new direction. So just to conclude, we have completed a very thorough population genomics assessment of Eucalyptus grandis, not just in terms of its fundamental niche, but also its realized niche in exotic environments also. I didn't have time to go into this very much, but we also related that population structure to environmental factors that could have contributed to what we see. We have begun to consider the effect that selection has on that structure. And the last step would be to refine and confirm some of these findings in common garden trials. And we have 2 large common garden trials going into the ground this year and next year. One is established by SAPBA. They're going to plant it in Zululand, and the other is going to be by Mondi in the Natal Midlands. And with this, we want to generate models for genotype by site matching. And because the natural range spans such a massive environmental cline, we can actually include more information in our models than we can get just from where the things are being planted in breeding trials. So I would like to acknowledge the Forest Molecular Genetics Research Group. That's us over there. We have fun. And of course, everybody that has contributed in some way. So it was a large project. So thanks.
Thank you, Maya. Do we have any questions for Maya?
Dave. Oh, Dave.
Thanks, Maya. So you've identified what you think is a selective sweep there. So do you think that came about due to a decision made in that early South African breeding program before the companies got to it? Or do you think it's something which is somehow developed after that?
I think it's something that, it's hard to say. It's probably something that came in before they split them into the 3 different breeding programs. And the reason why I say that is there's another peak that we investigated, and that is differentiated among the 3 programs. So it's clear that there was something where one program selected it preferentially over the other. And the other reason why I'm saying I think it's something that came before, or it's something that is just universally important in all 3 programs, is because this material was sourced from different areas. The Marensky material was sourced from Sanin. It was bred in Sanin for, I think, a couple of generations. The SAPBA material was sourced in Zululand. It was bred for Zululand. But that's only 4 generations, 3 generations. So I think that's probably something that happened earlier, but it's still important now, which is why it hasn't fizzled out yet.
Is there something special about the South African environment? And can you relate that to the 2 regions of Australia? In terms of what is being selected?
Yeah, so it's hard to say what it is about the South African environment because they're actually grown in very different environments in South Africa. It's not just a uniform environment. But there is something else that we do when we breed, and I don't know if we'll pick it up because it's actually relaxing natural selection. So when we plant seeds this deep in fantastic soil, exactly the right water, we coddle them. In nature, they fall on the ground and they have to root like it's no one's business. And we saw that when we brought the seed in. They root like crazy, the wild material, but the breeding material, they just don't make roots that quickly. So I think we're relaxing some natural selection pressure that's in nature that we don't apply because we're nice to them. But I think there's also, so there might be breeding practices that we unintentionally also put selection on. So it's a very complicated picture. I don't think we can relate it at this stage to a particular environmental factor or anything like that. It's a very complicated picture.
Oh, we have one more question.
Up there. Am I looking at phylogenetic analysis? Within the species? Okay, I think the best resolution we're getting is with the principal component analysis in combination with a structure-like analysis. They're all giving us the same answers. The differentiation between the breeding and the wild material is around the same level as the northern wild population from the southern wild population. So it's that different from each other. We have definitely made some shifts. And when we look at a PCA, which you don't always get with a proper phylogenetic study, is I expected, because we have material from the north and material from the south, I expected the breeding material to fall in between the north and the south, like intraspecific hybrids, but they're actually grouping away. So it seems like we've made shifts that's not just mixing genetic material.
Okay.
Good.
Thank you, Maya.
Thank you.
Okay, so our next presenter will be from UKZN. It will be Kwame Shamorera. He will be talking about analyzing traits among wheat for drought and non-stress. And
Good afternoon, everyone. I'll be presenting on combining ability, taking you back a little to conventional breeding. Among F3 lines for drought tolerance, in wheat, under drought-stressed and non-stressed conditions. So we all know that production is being hindered by recurrent drought, especially in the country, and drought is affecting wheat production in South Africa. So there is need to develop cultivars that are more drought tolerant in wheat production. And ARSC, the Small Grain Institute, together with UKZN, have been collaborating to develop new drought tolerant cultivars for the South African market. Because we found that most South African farmers in wheat are experiencing a lot of trouble and profitability of the crop is really reducing. So there is need to produce drought-tolerant wheat for the farmers. So the purpose of this work was to determine the combining ability of selected lines under drought-stressed and non-stressed conditions. So this project actually was initiated in 2014 by Mwadzingani et al., and he started the initial process of selecting the different cultivars the different parental lines under stressed conditions. Then he selected some, then he crossed them, then he advanced them. But because we wanted to accelerate the breeding process, we decided also to do a combining ability analysis at F3. So the objectives of the study was to estimate the combining ability effects and the mode of gene action on yield and yield components, and to identify families that we can select at early generations. So we did 2 experiments. So the first experiment was in the greenhouse. The total amount of genotypes that were evaluated was 78. We had 12 parents that were selected initially, and when they were crossed, we advanced them to the F3 families, and then they were evaluated together. They were evaluated in a half-diagonal mating design, and there were 2 water conditions. There was one condition where we ensured that there was sufficient water, and the other one, we imposed drought stress at 35% of field capacity. So we tried to target our stress at flowering. So after 5 weeks, that's when we turned off the water. So this is one of the greenhouse trials that we had in the greenhouse, and you can see from the picture that this side, that's where we imposed the drought. So you can see there's a difference even in the maturity of the crop. So the other one was conducted in the field, at Ukulinga Research Farm. So we used a black plastic mulch to cover up the soil surface to ensure that no rainwater would enter into the soil. We also did the same in the field trials we did in the greenhouse. We imposed stress after 5 weeks of planting. So this was the field trial. So we collected data on all the agronomic traits, especially focusing on shoot biomass and grain yield, and also days to heading and maturity. For our analysis, we did an ANOVA, and we separated for the diallo analysis for all the 4 test conditions. We used ARA, the HD-ARA system, which is commonly used by CIMMYT, and the estimates of GCA and ACOA were done using the diallo method 2, model 1. So from our results, we discovered that there was quite significant GCA effects that were observed for shoot biomass, grain yield, and days to heading. And then also under non-stress, shoot biomass also could be seen that there were significant GCA effects as well. So we can see that there was quite an influence of additive gene action in the inheritance of those respective traits. Then SCA effects were also seen for days to heading and days to maturity. And under stress and for shoot biomass and grain yield under non-stress, signifying non-additive gene action. The heritability for all the traits was very low. It was below 50%, but it was something that we were expecting since they were progeny at the F3, so there was quite a lot of segregation that was still in the plant material. So the phenotypic expression wasn't a very good representative for us of the gene effects that the wheat had. Then the Becker's ratio also was below a unit for all the traits, which showed the preponderance of non-additive gene action. So the combination of both low heritability and the predominance of non-additive gene action complicates selection. So that's why we were targeting to use early generation selection so that we might be able to fix whatever we get by using double haploid techniques to fix the homozygosity at F3. So focusing on the actual lines that were being selected for, we identified LM17, which had negative GCA effects for days to heading and maturity, and also a negative GCA for plant height, which is desirable for us when we are breeding for drought tolerance. Then also positive GCA effects were observed for those following traits for the respective lines, with LM13 and LM23 and LM2 showing significant GCA effects for grain yield. So this was just a small table to show in a graphic representation to see that LM17 had a negative GCA for those traits. So we can focus for we can focus to target for early escape, for flowering, and also for shorter genotypes. And then for the other traits, especially LM2, 13, and 23, there's a positive GCA for grain yield, and we can target that for improvement of the grain yield. Then for the actual families that were crossed, we identified families LM2 and 5, and LM2 and LM17, which had high yields. The reason why we decided to identify these ones and select them, for instance, is that both parents had high positive GCA effect for yield components, and also they maintain those GCA effects over over the different environments. And also we found that these specific families also had a high SCA effect for grain yield, and we recommend these specific families for genetic advancement, and also to fix for homozygosity and for multi-environmental trials. So in summary, these were the lines that we identified, LM2, LM13, and LM23, which had a positive GCA effect for grain yield under drought. Then LM17 can be used to develop early maturing dwarf genotypes, which will be targeted for drought escape. So these can be used in other breeding programs. So we can select these families and develop double haploids to fix the genetic makeup of these families, and also use the selected lines for developing new breeding populations. And currently there's a project that is also running that is now focusing on root characteristics, and some of these lines that we selected were used for that purpose, to develop these lines for drought tolerance and improved carbon sequestration. Would like to acknowledge all these partners that were involved in the work, and thank you for your attention.
Thank you, Kwame. Do we have any questions? Any questions? Thank you. Thank you very much. Okay, next up we have Fenele Msani. She's also the awardee of our travel grant, and she will be talking about sugarcane research. She's just getting her mic ready quickly. Okay, good luck, Fenele.
Good day, everyone. I'll just present my short presentation on the topic which says the effects of spacing, which is trimline, and continuous spacing on genetic and breeding parameters of sugarcane population. The South African Sugarcane Research Institutes have 7 sugarcane breeding programs. In these breeding programs, they all undergo 4 sugarcane breeding stages, which is seedlings, mini lines, single lines, and variety trials. These stages uses different row spacing. The first stage of field evaluation uses trimline spacing, while single lines and variety trials uses continuous spacing. This presentation or this study will only focus in the first stage of field evaluation, where trimline spacing is used. One may ask, what do I mean by trimline spacing? Trimline spacing is whereby 2 adjacent planted rows are followed by the unplanted row. The 2 adjacent planted rows are spaced using continuous spacing, which is equivalent to 1.2 meter. And trimline spacing is equivalent to 2.4 meter. These pictures here shows the difference between trimline and continuous spacing. The below picture shows the first stage of field evaluation, where trimline is used and continuous spacing. And this picture here shows the other stages, the mini lines and variety trials, and also commercial planting only uses continuous spacing. Trimline was designed to give extra space or extra access for data collection, disease inspection, and visual assessment during selection. However, the effect of trimline on sugarcane population is not known. Therefore, the study wants to determine and evaluate the effects of trimline spacing and continuous spacing in the first stage of field evaluation, as where families are selected for the next stages. If the bias do exist, if the biases do exist or is significant, that might affect the selection efficiency and the strength of a breeding programs, because the other stages, the following stages, only uses continuous spacing, not trimline spacing. So the effects of row spacing have been reported and show that spacing affects growth and development of sugarcane plants, and also a significant of variety by row spacing interaction have been reported, which means that when two or more varieties are planted under different row spacing, their performance differ across row spacing. And that concerns us because if two or more varieties are affected by spacing, two or more spacing, how will the population of sugarcane families be affected by trimline and continuous spacing, and how will that affect our selection efficiency of superior families? Which brings us to the objective of the study, to determine the effect of spacing, which is trimline and continuous spacing, on variant components, broad sensory readability, and genetic coefficient of variation, and also the implication on family evaluation and selection. Trial was planted at Mpangeni Research Station using a strip plot design, and there were 20 families in total, replicated 3 times, and there were 10 genotypes nested within family plots. And there were 3 experimental units. I'll just show you the experimental units now. This is the typical example of a strip plot design that I will show you now. So F1 to F5 are the families, T and C are the spacing used, which is trimline and continuous spacing. So each family goes across each spacing, and each spacing goes across each family. And there is spacing by family interaction. And these are the 3 experimental units: spacing, family, and family by spacing. Data was collected at crop maturity with stock numbers. All the millable stalks were counted, and with stalk height, it was measured from the ground to the last leaf collar, and the digital caliper was used to measure stalk diameter. And this formula here was used to calculate our cane yield. So we analyzed our data using SAS software, using this statistical model to generate our variant components, and from the variant components, broad sensory readability and genetic coefficient of variation were calculated using these formulas. If we looked at our results, the result that we got shows that spacing, the family variance component was significant for all traits across spacing, except for cane yield under continuous spacing, which might suggest the potential bias caused by continuous spacing or spacing. And also the significant family variance suggests the presence of family variability across spacing for these other traits. And we wanted to look at the magnitude of the family variant component. We wanted to see how trimline and continuous spacing affects this component. And we calculated trimline to continuous percent for variance component, and the results that showed. The results came out, and it shows that cane yield and stock numbers had higher values under trimline as compared to continuous spacing, while stock diameter and stock height had higher values under trimline than continuous spacing, which also suggests the potential bias, because if we select for higher cane yield, more families will be selected under trimline spacing as compared to continuous spacing. However, the next stage uses only continuous spacing, which means that families might not possess the similar values when planted under continuous spacing. We also looked at the family by replication and residual error variance, and also we calculated trimline to continuous percent in order to see or to determine how spacing and how trimline and continuous spacing affect these components. And the results show that family by replication had higher value. Family by replication was higher under trimline compared to continuous spacing for all traits, which might suggest that families varied more across replication under trimline spacing as compared to continuous spacing. These results may also suggest that predicting family variance will be lower when trimline spacing is used, which also creates a confounding effect. And the residual error was higher under trimline spacing than continuous spacing for all traits, suggesting that the variability was not accounted for by the design under trimline spacing as compared to continuous spacing. So after we generated our variance components, we calculated percent heritability and genetic coefficient of variation, and the results also showed a similar trend as the family variant component. The results show that tramline had higher percent heritability and genetic coefficient of variation for cane yield and stock numbers, while stock diameter and stock heights had higher values under continuous spacing than tramline spacing. These results suggest that also if we do select for higher cane yield, more families will be selected when tramline spacing is used, which might affect the selection, or we might not select the superior families due to the confounding effects or the bias caused by tramline spacing. However, the higher heritability and genetic coefficient of variation for stock diameter and stock numbers, those were the good results, because if we do select for these traits, it means that when we planted those families and continuous spacing for the next stages in commercial planting, they will also possess the similar values due to the spacing that is used. So in summary, as I have mentioned, that tramline increases family by replication variance, which might require more replication under tramline as compared to continuous spacing, and also that spacing affected traits differently. For example, higher percent heritability and genetic coefficient of variation for cane yield and stock numbers under tramline as compared to continuous spacing, and higher percent heritability and genetic coefficient of variation for stock diameter and stock height as compared to tramline spacing, which highlights the importance of understanding trait dynamics in sugarcane breeding, and also the differences of cane yield, of heritability and genetic coefficient of variation suggest a potential bias associated with different traits. However, adopting similar spacing across sugarcane breeding stages might minimize the potential bias caused by tramline spacing in the first stage of field evaluation. The takeaway message is that spacing does affect the population of sugarcane families, and that might affect our selection efficiency and the strength of a breeding programs. And more studies need to be done in order to validate all those results. For example, calculating the correlation, the genetic and phenotypic correlation between traits under tramline and continuous spacing. I would like to acknowledge the following organizations and my supervisors. Thank you.
Thank you, Fanele. Oh, we have our first question.
Thanks. To what extent do you think the accuracy of putting in tramlines affected your CVs? Because, you know, with regular crop plants, planters, should I say, we have an issue with when you turn around the planter and you come back. So if you have a 4-row planter, the rows are exactly replicated every time, but there's a problem where you come back with the planter and it's that intermediate row between the two planters that is perhaps not that accurate. And so what I'm asking is, how accurately do you put in the tramlines? And could some of this variation that you see is actually your row width varying because you don't have a good marker or something like that?
I think the variation that we experience is due to the extra space under tramline spacing is it equivalent to 2.4 meter, because is equivalent to 2.4 meter. And the families, they do not compete for growth resources such as sunlight and other growing resources, as we have seen that the family variability was higher under tramline for stock numbers compared to stock heights under continuous spacing. Continuous spacing, there's a lot of competition between families, so the higher variability for stock heights, for example, was due to the limited space and due to the competition among families.
Thank you. Do we have any more questions? Okay. Thank you, Fanele.
Thank you.
Okay, next up we will have our flash talk presentations. So each presenter will only have 5 minutes to present their research to us. To make it more interesting, we thought we might offer a prize to the winner of this flash talk. So underneath your plant, you will see a little voting paper. So if you might just vote for your favorite presenter, please. And then afterwards, we will have a box in front where you can place your votes. Our first presenter will be Annerie. Thank you.
Can everyone hear me? I assume we're fine. Okay. So I'll be presenting on using a phased genome assembly approach for Eucalyptus europhylla, Eucalyptus grandis, F1 hybrid genome towards enabling haplotype-based molecular breeding in eucalyptus. And the main aim of the project is to produce a high-quality reference genome for both Eucalyptus europhylla and Eucalyptus grandis to enable this haplotype-based molecular breeding strategy. So the reason we want to look into this is because haplotype-based molecular breeding strategies allows for the selection of gene variants instead of looking at individuals, markers that can be selected. And this is a much more effective and accurate way of improving traits that are being selected for and doing it in a better way. But the basis of this strategy is making use of a reference genome. And although we do have a reference genome for Eucalyptus grandis, we currently do not have a reference genome for Eucalyptus europhylla. And it is, unfortunately, the grandis reference genome is quite fragmented, and it represents a mixture of the two haplotypes or chromosome sets that were present in the individual that was sequenced. And this is partly due to the sequencing technology that was used. But we now have new long-read sequencing technologies, and these offer a variety of advantages. But what we're interested in is the ability of these long reads to span across haplotypes, and it allows you to separate out those haplotypes and assemble those independently. And if you have a hybrid, F1 hybrid between two species, you can assemble a reference genome for both species. And to do this, we are proposing using this trio binning approach that Corin et al. Also used in cows, but it worked, for assembling reference genomes for two species. So what you do is you do long-read sequencing for the F1 hybrid, along with short-read sequencing for the parents, and then you use the short reads of the parents to separate out and bin the haplotype or chromosome pairs, if you can call it that, but they're not that long. In any case, you can separate these out into separate bins corresponding to the short reads, and by using the short reads. And these you assemble independently, and then you should be able to get genomes for both of these species, which you can then use for comparisons, start looking into structural variation between the two species, and these variants might be linked to species-specific traits that are of interest to the breeders. So towards this, we've already performed nanopore sequencing for the hybrid, we've done the Illumina sequencing for the parents, and we've successfully been able to separate out the reads between the two bins, and these are currently busy assembling. So to conclude, I'd just like to sum it all up is that this long-read sequencing strategy will allow assembly of high-quality reference genomes for both of these parental species, along with using this trio binning approach. And towards this, we've generated 67 gigs of nanopore sequencing data for the hybrid. So this is about 50 times coverage per parental haplotype, which we've been able to bin and separate. These phased genome assemblies are in progress, and these reference genomes will become a starting point for using this haplotype-based molecular breeding strategy, where you can select for gene variants instead of single molecular markers. Thank you. No question.
Thank you, Henri. Unfortunately, with these sessions, we won't have time for questions, but afterwards in the tea break, you can approach the speakers if you have a question. Next up, we have Mary.
Good afternoon, everyone. I am Mary, and I'm going to be presenting to you on whole genome sequencing of macadamia using innovative sequencing technologies for advanced genetic breeding. And this is part of my PhD project. So firstly, macadamia is endemic to Australia and is found on the east coast of Australia in the region between Queensland and New South Wales. My apologies. Okay, and commercial macadamia cultivars are composed of two species. That's Macadamia integrifolia on the top there, and Macadamia tetraphylla. However, initial selections for macadamia were done in Hawaii, where they selected mostly from the integrifolia species, which selected for a high oil content, so that it enabled easier roasting as a confectionery product, whereas in South Africa, the farmers who initially brought it into the country preferred the sweeter taste of the Macadamia tetraphyllas. Hence, most of these cultivars that you see here are those composing of the local selections done in South Africa. However, South Africa is mostly planting commercial cultivars that had been released from Hawaii. So in terms of production, South Africa is the #1 producer of macadamia in the world. That's that green bar there, followed very closely by its native homeland, Australia, which is the blue since 2011. So South Africa is leading production of macadamia in the world. And in terms of quantities, South Africa in 2018 had an estimated 60,000 tons of macadamia being produced. So for the 2016-2017 financial year, the South African macadamia industry was valued at around 3 billion rands, and this just shows you how large that the industry actually is. But most surprising to note is that there is no formal molecular breeding or genetics breeding that is currently being done in South Africa. So we have no way of genetically differentiating between the different cultivars, so everything is based on phenotypic data. And added to this, macadamia genetic resources are very limited, with the reference genome only coming out in 2016. So this clearly tells us that if South Africa wants to remain above and be the top role player of macadamia internationally, there is an urgent need to include genomics in advancing genetic breeding for macadamia. So with this, we are using two genetic tools, which is microsatellites that can be easily used to screen cultivars and genetically differentiate different cultivars, as well as give us information such as genetic diversity and use for parentage analysis. And our second genetic tool is using sequencing technologies. So using microsatellites, this is just a subset of the cultivars that we have, and we see that the microsatellites we're using here are those published by Langdon 2019, and they were able to successfully differentiate between cultivars as well as between species. So in the green, we have the Macadamia integrifolia cultivars. In the red, we have the Macadamia tetraphylla cultivars, and in the pink is the known cultivars that we are aware of, and macadamia does naturally hybridize in the wild. And we were also able to identify 791, which is a hybrid between Ternifolia, which is a third macadamia species. So clearly we can see that microsatellite markers are useful in differentiating between cultivars and definitely can be implemented in macadamia breeding programs. So moving on to our next genetic tool, which is sequencing. And with that, we are using the current gold standard of genome assemblies, which is a hybrid approach combining different sequencing technologies to assemble the most concise genomes. So we're using short-read Illumina sequencing combined with long-read Oxford nanopore sequencing. And together with that is the bio-nano optical mapping, which will be able to let us zoom into the genomes. And for that, It'll be able to help us with the early flowering, improving yield and nut quality for macadamia, as well as decrease breeding time, and obviously with all the talks we heard yesterday, climate smart agriculture. So finally, whole genome sequencing is a necessary investment for crop improvement and climate smart agriculture. With that, I would like to acknowledge the following people and institutes. Thank you very much.
Thank you, Mary. Next up, we have Julia.
Good afternoon, everyone. Today I'm going to be giving a very brief overview of my master's study, which aimed to determine the feasibility of using a multi-parent mapping approach for QTL mapping and analysis of segregation distortion within eucalyptus. So I had two main aims. The first one was to identify genomic regions and DNA markers for molecular breeding, and secondly, to dissect the genomic basis for hybrid combining ability. So the multi-parent population was constructed by SAPI by crossing 9 Eucalyptus grandis pollen parents with 8 Eucalyptus europhila seed parents. This resulted in a large number of full subfamilies nested within half subfamilies. As this was just a test of feasibility, we selected 1 grandis half subfamily and 1 europhila half subfamily, and the numbers inside the trees represent the number of progeny we had per full subfamily. Additionally, the population was planted across 4 different sites within South Africa, which allowed us to then determine or perform our analyses across the entire population and also within each of the individual sites. So we first had to construct genetic linkage maps using SNP markers, and then after this we mapped QTLs controlling growth and wood properties. So we first did this across the entire population, and the green blocks represent some QTLs that we did identify. We then mapped QTLs within each of the individual sites, represented by the different colors. And what we found was that we did not identify any QTL that was found in the same location across both sites, or 2 sites. But this could just be due to the fact that we didn't have a high enough power. So we cannot conclude any genotype by environment effect here. But what we can conclude is that the multi-parent mapping approach can be used to dissect quantitative traits in eucalyptus. Next, we analyze segregation distortion. So if our pollen parent is heterozygous for alleles A and B, we expect to see both alleles in equal frequency within the progeny. However, sometimes you see that one allele is favored over the other, and this is segregation distortion. And we can visualize this using this graph. So in the graph, each bar represents a marker, and if it's below 0, it means the one allele is favored, and if it's above 0, it means the alternative allele is favored. The red line represents the statistical test, and the gray line represents our critical value. So basically, red shaded regions represent regions of significant distortion. So this graph just represents the segregation patterns for the Eucalyptus grandis pollen parent alleles across the entire half subfamily. What we then did is analyze the segregation patterns across the individual full subfamilies. And what we saw was there was a large amount of variation in the patterns of segregation distortion across the chromosomes and also across the different full subfamilies. But the population was planted across 4 different sites, so we needed to take into account the site effect. So what we first did is we analyzed one site with many full subfamilies present on it. So the bottom graph represents the segregation distortion for the site, and the top 5 graphs represent the segregation distortion for the individual full subfamilies. And again, we see varying patterns of segregation distortion. Additionally, what we did see is that some full subfamilies had one allele favored, while a different full subfamily had an alternative allele favored. We then analyzed one full subfamily that was planted across different sites to determine this environment effect on segregation distortion. Again, we saw varied patterns of segregation distortion, and interestingly, we saw that one allele was favored on one site, while the alternative allele was favored on the other site. So what we can conclude from this is that segregation distortion is caused by parent-specific interactions which depend on the environment. So with that, I'd just like to acknowledge my supervisor, the FMG Research Group, and my funding bodies.
Thank you, Julia. Our next presenter will be Kaneo.
Thank you once again, ladies and gentlemen.
I'm just going to give you an overview of my PhD study, part of my PhD study, entitled Proteomic Analysis of Abiotic Stress Influence on durum wheat gluten protein composition. Despite climate change, high grain quality is essential for the durum wheat cultivars to be accepted by the industry. Gluten protein, a component found in the grain of wheat, is responsible for grain quality. But the problem is gluten is dynamic, may change in response to changes in the environment. This may in turn affect the grain quality. Drought and heat are the major abiotic stresses which influence grain quality. Therefore, if we are to improve grain quality, it is of great importance for us to understand the influence of the environment on gluten protein composition, as well as to identify the proteins that change in response to environmental changes. And this will help us to predict functional end-use quality. You know, quality is what the manufacturers, the consumers need. In this study, the aim was to assess the effects of the environment using two-dimensional gel electrophoresis. This technique is able to separate the complex mixtures of proteins, as well as to identify those proteins that change in response to environmental changes. So in this study, we used 2 durum wheat cultivars, which differs in terms of their quality characteristics. The other one was a good performer, and the other one was a poor performer in terms of the quality characteristics. So these cultivars were planted under various stress conditions. So the results obtained from this study indicated that in one cultivar, Mexicali, which was a poor performer, moderate drought stress had a large influence in increase in proteins. While in Artil C, a good performer, the increase in proteins were due to various stress conditions, were due to all stress conditions except for control. And with the proteins that were identified fell into 5 categories, being the high molecular weight, the gliadins, the serpins, the globulins, and the alpha amylase. These proteins have been linked with quality. The other thing that we can say about in this study is that drought and heat really had an influence in gluten protein composition, therefore should be taken into consideration. Remember, we eat not just to eat, but we also eat for enjoyment. Thank you.
Thank you. Our last flash talk presenter will be Tulo.
Greetings, everyone. I would like to open with a statement that says genetic diversity is a foundation for plant breeding, and therefore it's a breeder's job to develop varieties that are, or rather that can withstand the ever-changing climate conditions, but of course still keeping the good yields. Now, with that being said, the topic of my talk is characterization of sorghum germplasm using both phenotypic and molecular markers. Sorghum is one of the most important cereal crops in the world. It ranked 5th after maize, rice, wheat, and barley in terms of both area planted and production. The crop is indigenous to Africa, and it serves as the staple food source for millions of people in Africa, Asia, and the semi-arid regions worldwide. It also has extensive root system and a C4 synthetic nature, which allows it to grow even in arid regions with poor soil fertility. Now, global warming often leads to an increase in combination of both biotic and abiotic stresses, which then affects yield and production of major crops like maize, rice, and wheat. This in turn results in reduced food supplies. In contrast, sorghum as a more drought-tolerant crop is less affected by these climate changes. With that being said, it is therefore in important need to characterize the sorghum accessions to to basically come with accessions that can withstand these ever-changing climate conditions. The main objective of my study was to then characterize or evaluate genetic diversity among ARC sorghum germplasm using both phenotypic and molecular markers. 100 sorghum accessions were used in the study, and the phenotypic, the phenotyping was done at Potchefstroom over 2 seasons, and it comprised of 14 quantitative and 5 qualitative traits. The actual genotyping was done at LGC Genomics in UK, and it comprised of 136 competitive allele-specific PCR markers. These markers are also the SNP markers. They are converted by LGC Genomics for in-house genotyping. Now, as shown in this in the slides there, under phenotypic data, there was wide variation in terms of the qualitative traits. In figure 1 is just the variation in terms of the panicle shape. Now, the open panicles would be ideal in humid areas that receives high rainfall because it basically makes them less vulnerable to diseases such as grain mold, as compared to compact panicles, which would be ideal in arid regions where diseases are not severe. Figure 2 is just the variation in terms of the midrib color, in which the green midrib color indicates the stems that are juicy. White midribs indicate stems that are spongy, and brown midribs are associated with highly ruminant digestion rate and can be used in livestock feed. Under genotypic data, figure 3 shows the accessions being clustered largely in terms of their breeding status and geographical region, and they were grouped into 3 major groups, in which cluster 1 had only 4 accessions, cluster 2 had about 27 accessions, and it was dominated by the R lines from the USA. Cluster 3 was the largest with 64 accessions, and it comprised of B lines, R lines, and some land races. The maximum genetic diversity, genetic distance, was observed between the accessions, SA 1442 and Hakika, meaning that they share very different background and can be used in breeding. In conclusion, the accessions showed a wide genetic diversity, which could then be used in breeding programs to further secure food security. Thank you.
Thank you, Tilo. Thank you, everybody. That marks the end of our session. Can we have one round of applause again for all our speakers, please? Thank you. Okay, we will have a tea break now until 3:20, and then we will continue with the session. The valid
Can you tell us how to do it? To rank them or to give them a vote?
No, you can just make a tick next to your favorite presenter. Yes, one tick. Yes, thank you. Yes. Yes. Is it clear to everybody how to vote on the voting ballots? You give one tick for your favorite presenter. Yes, only one. Thank you. They're coming back, I think, for the other session.
Hello, sir. Can you hear me? Yes, can hear you loud and clear. Can you just please put your screen share on for us? You just share your desktop and then you can just open up the PowerPoint on your side. Um, just hold on. Yes, we can see you. If you can enlarge it for us, just quickly, we just want to see the side. If you can go into PowerPoint full screen. Okay, we're cool. We're getting everything that we need. Well, I'll send you the microphones on this side if anyone has questions for you. And then we'll be good. I think we'll give you the go ahead on when you can go. If you can just be on standby for us. Okay, we should be going live in about 5 to 10 minutes. So if you can just stand by for us on that. Thank you.
So good afternoon, everyone, and welcome to the final session of the day. So we will be having one presentation by Emerson Limburger, and this will be televised up on the screens. So he is based in Iowa, USA. He's a research scientist at Corteva Agrisciences, and he's the coordinator of the molecular breeding activities for Europe, Africa, and the Middle East. So after this presentation, we'll be taking some questions, and we will then be moving on to our poster presentations. So enjoy the session. Great.
Okay. Good afternoon, everybody. My name is Emerson Limburger, and together with Andrew Danner, Dave Spalding, and Carla Vogel, we coordinate a group that is responsible for delivering all of the genetic information for the company within Corteva. So basically what we're doing is collecting all of the SNP data that is generated across all of the crops and all of the geographies, and we are delivering that for end use in terms of genetic predictions for breeders or marker-assisted selection for all of the lines that are going to be going into CRISPR or for transformation. All of this genetic data we have to receive and transform this information into a single genetic profile that is available for all of the company. So that is pretty much aligned with this concept of big data. So I'd like to thank the Southern African Plant Breeding Association for the opportunity for us to talk here today, especially Drian and all of the students that enabled us to belong in this Plant Science Symposium series. So moving forward, I'm showing here a graph that shows the trend in the world population. So the world population is growing, and one of the goals for us as plant breeders is to feed the world. So with the world population growing, we have to make crops be delivering higher yield, able to maintain the world population being able to feed. And in comparison to that, I'm just putting the trend line that shows what the data point in terms of SNPs is since 2004. And the reason I chose 2004 is not only the reason that it's when we started to work more heavily with marker data, but it's also because that was the year in which I started working at the company. And then you can see that the trend is a much faster growing trend than the world population, which give us a little bit more of headache in terms of like how we're going to manage all of this information at this growing rate. So there are new genotyping technologies. So in the past, you would have assays that would deliver very few markers. Now assays are delivering more and more markers within the same set. So that is helping increase this volume. There's also higher lab throughput. Some of the reactions that sometimes would take a few weeks, now they take a few hours. There's also a lot of investment by the company delivering labs in different geographies where you're doing plant breeding, and that includes Delmas in South Africa, and also the adoption of markers by the breeding in different plant breeding stages. So in the past, there were very few steps where markers were being used, and now we can say that the markers are being used pretty much in every single step in plant breeding. So that makes the volume of data increase gigantically. So I'm just showing here as parameters the rows in Excel. They're very insignificant with the amount of data that we have, and also the milestone that we accomplished in 2014, which is reaching the corn genome size in terms of SNP data points, and then trying to align as well with the challenges that we have, and is the focus of this mini series here. The world population is also the world average temperature is also increasing. So global warming is a fact. Because I started in 2014, that's not too obvious. But if you go back a few more years, you'll see that that's a very strong trend, especially in the most recent decades. And we know that the world temperature is rising because of human impact. We are increasing the concentrations of several gases, and CO2 is one of them. And those emissions are retaining the temperature on Earth, and that's why the temperature is increasing. So with this rise in temperature, we're going to now have an extra challenge within our scope. So we have to feed the population that is growing, but we also have to make sure that those plants that are increasing in yield, they're also going to be adaptable for the new situations that those plants are going to be grown. So droughts are more frequent, heat stress as well, and how we're going to deal. And that's when we think that the big data is going to come here to help and support make those decisions more efficient. So as I mentioned before, and there was a presentation already today with wheat drought tolerance, this is going to be critical in the future. Heat stress tolerance, and we think that whole genome prediction are tools that really are going to help the breeders to move forward with better selections in faster selections that they would do in the past. There's also insect tolerance that can be controlled by transgenes. This is a very, very strong reference already. There's disease resistance that can be managed with CRISPR. And both insect tolerance and disease resistance, they really help crops to deal better with those challenging circumstances of drought and heat tolerance when they don't have to deal with extra stress. So on the left here, you have the average world temperature as it used to be with a representation of its population. And now what the world temperature looks like today, the increase in temperature looks today, and also the increase of population. So we also seen today some of the recombination events in eucalyptus. And here is the exact same concept that we've seen earlier in some of the sessions. I'm showing here how the impact of increasing the data in marker density is going to help us with a better IBD. So IBD is identity by descent. And if you look at those bars, they all represent different chromosomes, and each one of the color codes is going to represent the inheritance for one of the parents within the population. And then you can see different recombination events. But you can also see that I have highlighted here two boxes, a blue box and a red box. And you can see that when we move from low density to mid density, you have pretty much the same result. But you can see already on the 10th chromosome, where you see in red, that our probability has enhanced towards the blue parent. But we don't see any change in the chromosome 6, where it retains as yellow. As we move it into a higher density, now we can see that there was actually a double recombination event that was very narrow, but we couldn't capture that in a lower density. And then if you move forward to the other chromosome, you can see the exact same pattern. As we go to the very high density, we confirm that those events were true, and they were meaningful. So now that we're using this framework of IBD to make our estimation set and the prediction targets orthogonal in terms of the markers that are behind those predictions, we can see how this is going to affect the GBV, or the genomic enhanced breeding value. So I have 3 different traits here: dry matter, starch, and yield silage. And then you can see how it changes as we change those recombination events, as we had originally from a lower to a higher density. And that's going to be critical if you think that some of those selections, they're going to be very narrow performance between different individuals, and you want to make sure that you're making the good selection. So again, going back to the concept here is really the paradigm. We want to increase the data, but we want to keep the error low. And how is the marker density really affecting behind the scenes in a prediction scenario? So now what I have generated here, I have captured a mean locus effect from an estimation set, and I took that And I transferred into the genetic profile of every one of those individuals. And what you can see here is that you have different effects. And if you look at this marker 1, it has a negative effect for a given region of this chromosome. But then when we increase the density, so remember we're going from a low density to the high density, we see now that this effect is changing to the positive effect. And the reason for that is because we're actually estimating the incorrect parent within that region because we're not capturing the double recombination event. And that's how the bigger volume of data is really going to enhance us and give a more powerful way of making selections in this whole genome space. So doing that for several years, we have realized that the control of the data that we were having before was not enough. So here you have this chart that is showing the canola genetic map, where every single pixel that you see is going to represent an individual in a chromosome position. So you have the chromosome as the column. So you start at chromosome 1 and you go all the way to chromosome 19. And then as you go down, you're going to have from entry 1 until the very last entry. And now we have the low density, and how what we're seeing here is actually the causative matrix that is used both in the estimation set and the prediction targets. And what we are seeing is that everything that is in red are markers that have not been genotyped, and we have failed to impute, which are the markers shown in blue. And then everything that has been imputed use the framework of those very few markers that we see in green. Okay. Now, when we move it into a higher density, what we realize is that we don't rely so much in imputation because we are basically genotyping all of the markers that we need to have for the estimation set and the prediction targets. But we still have a few of them that we need to genotype to impute. And now we also know that the imputation accuracy is going to be much higher than the one before, because we have a higher precision in capturing the recombination events with this higher density. And obviously there are going to be some of the markers that are not going to work. But in contrast from the low to the high density, you can see a very drastic difference. Okay. So we've seen some presentations today talking about genetic gain and heritability. So when we want to focus on genetic gain, a lot of people are putting a lot of efforts in the heritability, and we've seen how complex. There are some examples in sugar cane, how complex it is to have a correct estimate of our phenotypes, and how much this is going to impact the heritability. Now we're adding an extra component, which is this marker layer that is going to augment our estimations. So instead of having a purely general combining ability, you can have a predicted combining ability. But what we really want to focus here with this extra layer is that we have to keep the error down, because we already know how complex the heritability per se is. If we're going to add a new layer of markers and we're going to add error with this, we're not going to be very successful with our estimates. And this is really the challenge that this group has been working with, because we have come to the point that we realized just constructing those rules that is going to exclude a few individuals wasn't enough. So we had to go back and look at the data in the lab before it was coming into us. So we know that the IBD framework with a higher density is going to improve our ability to detect double recombination events. This is going to allow us to have a more precise way of estimating the effects and also assigning the correct effect for each one of the individuals. And we know that the higher density gives us a much better causative matrix for our prediction and estimation set. But how do we deal with the error? And this is really the challenge that we have, and how do we control it? So here I have, just to illustrate, again, when I started at Corteva, Office 2003 was out there with Excel, and there used to be 250 columns, and the Illumina Plex had already 668 markers. So you cannot open that in Excel as a solution. Now we're dealing with a much more complex case where Excel has 16,000 columns, but you can easily get Illumina chips with 56K markers. So the discrepancy is growing much faster on the genotyping side. That means, and the only reason why I'm showing this today is just to represent that now it's humanly possible for people to open the jobs and have a look and see if everything is okay before they go ahead and make the decisions. Nobody's looking at the data anymore. The data is just flowing and going into pipelines that is going to generate results for breeders to make selections. And that's where our error becomes a big issue. And we want to make profit of this big data, but we also want to control the error. And here as an example, so you've seen this curve before. Now we don't have the world population or the global warming trend. You only see the marker data point. But I have added a different layer now, which is the error rate. And the error rate is something that usually we don't know what it is. We can try to assess, but it's very hard to estimate what the error rate is. But the idea here is to look at the retrospective data and try to understand what the error rate is, and we know what it is. And as we increase the volume of data, we don't want the error rate to proportionally increase with that. So what we wanted to do is have the constant error rate, despite the fact that we have a greater volume of data. But remember that in the past, back in 2004, every single result from the lab was being looked by several different people from the lab all the way down to breeders. As per right now, with this volume of data, there's nobody in the lab looking at the results as they come, and there's no breeder looking at the job result, okay? And the challenge that I bring here is also, can we not only keep the error constant, but can we also decrease the error rate as we increase the volume of data? So this is the proposal that we wanted to show today. We are thinking about this convergence between the genetic data and the lab information, where we're going to come up with this diagnostic. So here you see on the left, on the top, the different nucleotides, and then you have on the right side a container from the lab. So what we're basically doing is what are the information that we have behind. So we have the dimensions from those containers, and then we can try to see, okay, we have different populations, and if each one of the populations we can color code it with a different color so we can identify them. And then we can take that genetic information and look, for example, for consistency with the parents. And now we have a heat map that's from green to red, and we can capture if there is a trend. And then if we can assign that and see which are the wells that fail and which are the wells that passed. And we can look here at a different matrices. This is not the causative matrix, but this is just showing the results as they come from the lab. So you're going to have, again, it's still ordered by chromosome and position, and then you have all of the different samples. And if you look very hard, you can see that there are different population structure, and you can see those by the recombination trends or the allele trends within each one of them. But when you look at the consistency here, so we're just changing the color code and now showing, okay, every time that it matches one parent, it goes with a yellow color, just like we had in the IBD framework. If it goes after the second parent, we color code in blue, and then everything that doesn't match the parents is a red color code. So then you start to identify issues with markers, you can identify issues with samples, and then you can finally identify issues with the entire populations that probably had a pollination error in the field. Okay, we can also look at concordance. So if we have retrospective data from those individuals, we can just see if it's matching or if it's not matching. And then we can use the same concept with color codes that are going to show us green if it's good, red if it's bad. And when it's gray, it just means that either we don't have reference or the parents are non-informative, or because the marker didn't work in that given job. We can also think of heterozygosity. So if you're thinking of a DH population, you wouldn't expect to see any heterozygosity. If you're looking at an F1 population, you'd expect to see a lot of heads. You can also see trends in markers or samples that are deviating from the rest. And then finally, data return. If there are an individual well that failed, if it has been caused during the sample stage, during the DNA extraction, during the transfer, during the PCR process. You can also see at the performance of different markers, if they're constant across all of the populations and all of the samples, or if they have different issues, and if we have to change any of the process. Okay, so putting it all together again, and bringing back the paradox, we want to have big data, but we want to have good data, because if we don't have good data, the amount of error that we're going to infer into the results is going to be huge. It's going to be very hard to capture. You won't know if that is part of the heritability or if just coming from the lab. And we want to prevent that from moving forward. So what we started to do is we changed a little bit the way that we're doing things here in the company. So in the past, we were detecting and doing the diagnostic at the same time. So we were running a lot of analyses that weren't necessary. And now we know that if we do the detection first, and then most of the jobs are going to be okay, and then there just pass a few jobs to specialized people within the lab that are going to be able to identify where the issue is coming from. So that enabled us to do much faster calculations and being much more precise from what we were doing before when we were trying to do both things at the same time. The other thing that we changed is that we were doing metrics and thresholds, and a lot of people really didn't understand where those thresholds and metrics came from. Some of them were just like legacy from one person to the other. The current manager wouldn't know. So we said, let's try to do trends, and the trends are going to be universal. And that's what we did. As most of our distribution, they are log normals. So now we're looking at heterozygosity. So we think that if you're looking at a DH population, you're going to tend towards 0, and then you're going to have a small deviation. And here we're looking at 4 different containers from the same job in a lab, and then we're seeing everything that is green is 100% homozygous, and as you start to go into yellow, you increase the heterozygosity until you're very hot in more orange shades. So we detect that. We know that sometimes we have issues coming from the field, so we drop those, and then in a trend concept, those disappear. But when the signal is very strong, then we capture that and we're going to fail. And that's when this is going to be sent back to the a lab personnel to have a look. And then in this case here, a plate sealer issue was detected. And we're going to, so try to remember this picture, because we will need that for the next slides as we move forward in an example of a detection case of a very similar issue, okay? So how does that happen? And that's why you don't need to have those thresholds. So imagine if you have the sealer and then you have samples that are homozygous A in well 1, and then homozygous C in well 2. If the sealer didn't do a very good job in closing those lids, you could have those samples contaminating each other. So now in both cells, what you're going to have is heterozygous AC, okay? And that's how we are detecting those issues. And then this trend analysis, again, is going to look at inconsistency to the parents, concordance to existing data, level of heterozygosity, and then we're going to make that diagnostic and color code them so it's easy for people in the lab to see where they happen and make a decision. We're also looking at other information like maximum neighborhood similarity, because again, when you're mixing 2 individuals together, they become like an F1 hybrid, a synthetic one, but they're going to share the exact same genetic profile. So if you find those very high identity, it's also an indication that wells have been contaminated among each other. okay? And then we also do some sort of correlation to determine for very specific types of chemistry and understand how those can be generated and how it can stop. And finally, we're going to run simulations where you can replace the genetic content from different jobs and try to see if it improves the performance in case some of the plates were swapped or shifted and so on. So going back to that trend analysis that I was showing before, here you have a field of double haploids, and then you have a set of lines that you're going to genotype, and the percent heterozygosity that you're expecting is around 0%. But then when the lab results come back, you see that you have a line that has 25% heterozygosity, and you have another line that has 6%. And how do you explain that? You could think that there has been an issue in pollination and discard those lines, which would be a huge waste of resources in the generation of those DHs as well as the genotype. And that's where we wanted to control this error and keep the data good. So now we're looking at 4 different projects that have been genotyped. So they're marked as 1, 2, 3, and 4. And each one of them is going to have 4 plates, plate 1, 2, 3, and 4. And we're going to look at those different genetic components. Now we're looking at heterozygosity. And then what we can see is that there are some areas of the plates that concentrates a little bit more heterozygosity than the rest, okay? And then we're going to run all of the concepts all together and assign errors for those, okay? So now we see that we have way more errors than what we were expecting from a head perspective. And that's where we have to, at the same time that we detect errors, we have to also control for errors that are not intrinsic to the lab, because otherwise we would be stopping more information that we would. And we also, when there's an issue that is originating in the field, we want to be able to give that information to the breeders to make decisions in terms of a forensics job that they can analyze and try to understand where the issue came from. So that is a valid data that we also want to deliver. So we go back and we start to look at different trends. So now we're looking at inconsistency. And then you can see that there's very high trends of inconsistency, especially on the project number 2, plate number 4, which is the entire plate is orange. There's also some issues in plate 3 and 1. So now it's where we add the pre-existing information, like population, for example, and then we see that we can correlate that with specific populations, which tell us that by no chance the lab would be able to generate this kind of trend that's originated in the field. So we could potentially pass. But we still have our original issue, which was the heterozygosity, and we have to solve that. As I mentioned before, if you mix 2 individuals, we're going to create a synthetic F1, and those are going to share the same genetic profile. And that's what we're seeing here now on the right side. And then the final step is when we're going to try to associate that. So I added, every time that we run on a specific equipment of the lab, which is a plate sealer, which is called plate lock EXT_1, and we were able to identify that we only had issues when we were using that specific sealer. Okay, so the remediation was that we could send somebody there to run maintenance in that machine and fix the issue, at the same time that we realized that we had to regenotype the individuals that were in those specific wells that were contaminated. Okay, and at the same time, the non-process issues, that means anything that is coming out of the field. So here again, we're looking at some DHs, and you can see that there are 2 very tall plants that are coming out of cross-pollination. So there was a contamination there, and that genetic information is not going to be able to fail the job, but it's going to be available for the breeder to understand which was the contamination that happened in the field. Okay. So again, big data, but good data. And this is how we're going to be able to come up with better solutions for all of the challenges that we have in plant breeding, and the challenge of keeping that error rate down. So again, looking at what is the expected data, what is the observed, crossing the information between the genetic parameters and the lab dimensions, and trying to capture these issues as they occur. And the final considerations, again, population is growing, climate change is changing, so temperatures are getting higher and higher. This is affecting rain patterns in different crop areas. And combined, they're going to bring more challenges for breeders. We already need to increase yield for an increasing population. Now climate change is going to bring that extra challenge. And big data is a reality in genotyping, so it's going to help us with the accuracy in imputation for our casual matrix that is used for GBV estimates. But again, paradoxically, it brings a lot of risks as we think of the volume of data that is being generated. It's humanly possible to manually review all of the data. So that's when the automated trend analysis can flag those specific jobs in a fashion that it becomes possible for people to do the intervention whenever possible. So here I finish my presentation, and if there are any questions, I will be glad to answer.
Thank you so much. Awesome. Great. Are there any questions from the audience?
So my question is about, in science, we're always looking for outliers and the exceptions. And with your system, if it's overfitted, are you not going to miss those important discoveries?
So it's always a challenge to make sure that we're not going to pass. One thing that we did to make sure that that we were not going to start missing good data was really to look back at retrospective jobs and see what we were going to do with those. But generally, I don't think that the way that we are applying right now is increasing the risk. I think that in the past, that risk was higher when you had a lower density. And one example that I use for that is when you think of the double recombination events, when we were estimating the IBD, and we would see an allele that wasn't matching the region in which it was a genotype, we were dropping that marker. So that was a valid, true information that we were missing because we didn't have enough information to validate that it was a true observation. But I agree with you. It's always very difficult and there are several of the controls that we do when we are looking at some of the changes that generally people ask us to do. There's always an impact on something else, and it's very hard to control.
Are there any other questions? Okay, great. Thank you so much, Mr. Limburger. Can we have a final just round of applause for the talk?
Thank you.
So now we will be moving on to our poster session. So just a quick note to the presenters to please remain by your posters throughout the whole poster presentation, just so that the judges can get you. If the judges get there and you're not there, your poster cannot be judged. And besides that, yeah, that is the final thing for today. The poster presentations will be taking place in the Research Commons. Okay, great. It'll be taking place in the Research Commons, which is across from this building. And just the final note to please take your plants with you as you leave. These plants are gifts for each of you. And then there's just a final announcement from Drian as well.
Thanks, well done. So an award from SciCorp for the presenter of the day, Maria Mostert O'Neill. Not yet? Yeah? She's gone already. Okay, we'll make sure this gets to her. Cool. Thank you. That's it. Enjoy the poster session.